Part 1. Antarctic sauropods. Long before human footprints marked the white continent, colossal sauropods wandered its high-latitude forests when Antarctica lay within the South Polar Circle, yet basked in temperate climates. Fragmentary bones and isolated vertebrae recovered from the late Cretaceous beds of James Ross Island hint at giant titanosaurs, relatives of South America's familiar saltosaurids, that thrived in ecosystems of towering conifers and fern understories. These remains, often distorted by glacial pressure and frozen for millions of years, reveal sauropods adapted to polar extremes. Growth rings in their limb bones record annual slowdowns during months of darkness, while richly vascularized bone tissue suggests bursts of rapid spring growth as sunlight returned to the horizon. The discovery of sauropod trackways on Seymour Island adds another dimension to the Antarctic story. Impressions of three-toed forefeet, trailing behind massive hind prints, tell of 5-ton beasts plodding across floodplain muds under autumn twilight. Track spacing indicates a deliberate, energy-efficient gait, perhaps a response to cool soil temperatures and slippery surfaces. The scarcity of overlapping tracks suggests that these giants moved in small family groups rather than sprawling herds, a social adaptation to limited polar resources where solitary wanderers had better chances of finding sparse vegetation than massive congregations. Eggshell fragments scattered across coastal outcrops pose tantalizing questions. Were titanosaurs nesting in the midnight sun, laying clutches on windswept plains some 65 million years ago? The microstructure of these fragments, with thick shells and minute pores, implies incubation strategies distinct from their southern cousins, possibly burying eggs in thermal vents or insulating vegetation to buffer against freezing nights. Without accompanying embryonic bones, the identity of the egg layers remains speculative, yet the daring notion of polar sauropods brooding in sub-zero air challenges assumptions about dinosaur reproductive limits. The largest Antarctic sauropod bone yet discovered, a single dorsal vertebra nearly a meter wide, bears deep gouges along its centrum. These scars may record predatory encounters with giant theropods, like the apex predator Cryolophosaurus, hinting at lethal struggles on ancient ice-free tundras. Alternatively, the damage could result from intraspecific combat, with rival males sparring with claws and neck muscles braced against the frigid wind. Whether these gouges reflect life and death conflict or ritual display remains an open question, one that underscores how much of polar sauropod behavior is tantalizingly just out of reach. Isotopic analysis of oxygen in sauropod bone phosphate offers glimpses of polar paleo-environmental conditions. Variations in isotope ratios between summer and winter growth rings imply temperature swings of up to 20 degrees Celsius, far wider than in equatorial settings. Such extremes would have tested sauropod physiology, maintaining core body heat in months without sunlight, likely required elevated metabolic rates or behavioral adaptations like basking on sunlit slopes. The interplay between physiology and behavior in these extinct giants presents a captivating puzzle in vertebrate adaptation. Antarctic sauropods also exhibit tantalizing signs of dwarfism in certain localities. Some limb elements, unexpectedly small relative to their cortical thickness, suggest island-like stunting, an evolutionary response to constrained food availability during long polar winters. This phenomenon echoes the polar dwarf concept seen in Pleistocene mammals, yet applying it to Mesozoic giants demands fresh thinking about resource allocation and energy budgeting in sauropods. How these enormous creatures balanced body size with survival in marginal habitats remains a critical mystery. Detailed CT scanning of Antarctic sauropod vertebrae has revealed internal cavities that likely housed air sacs extending from the respiratory system. These pneumatic structures would have reduced skeletal weight and facilitated buoyant balance on soft ground, but they also hint at high-efficiency lungs capable of sustaining activity in thin, cold air. Whether polar sauropods possessed more extensive air sac systems than their temperate relatives is unknown, but such an adaptation would have given them a physiological edge in the oxygen-poor, high latitudes. The very discovery of sauropods in Antarctica defied early 20th century expectations of dinosaur distribution. Early explorers dismissed reports of massive bones as misidentifications or transported remains. It took decades of careful stratigraphic work and international collaboration to confirm that these were indeed true sauropods, in situ, having endured polar cycles with little to no frost hardiness outside their own adaptations. This unfolding story reminds us that the fossil record, even in its most remote corners, continues to rewrite our understanding of dinosaur biogeography. Antarctic sauropods stand at the intersection of gigantism and endurance, giants born to roam forests under polar lights yet bound by the cycles of ice and sun. Each bone fragment, footprint, and eggshell shard carries whispers of ancient survival strategies, the rhythms of growth and dormancy, the boldness of nesting ventures, the scars of conflict, and the hush of long winters. As new expeditions reveal ever more secrets from beneath the ice, the saga of these polar titans grows richer, poised between the known and the unknowable in the frozen pages of deep time. Part 2. Ice Age Theropods. Antarctica's first theropod discovery turned heads when paleontologists uncovered the skull of Cryolophosaurus elioti in the early Jurassic snowfields of the Transantarctic Mountains. Aptly nicknamed the frozen crested lizard, this 2-ton predator bore a striking transverse crest above its eyes, the purpose of which remains enigmatic. Some propose it functioned as a visual signal in low-light polar conditions, while others argue it anchored powerful jaw muscles for dispatching hardy conifer prey or competing with rivals. The crest's bony composition, unusually thin yet resilient, hints at selective pressures unique to high latitudes, an adaptation still defying full explanation. Beyond Cryolophosaurus, fragmentary teeth and isolated bones suggest a diversity of carnivores roaming the high-latitude forests. Ceratosaur-like teeth, with hook-shaped serrations, imply smaller hunters stalking undergrowth, while larger, blade-like serrations point to apex predators. These finds, scattered across volcanic ash layers of James Ross Island, hint at a multi-tiered food web where theropods filled niches from stealthy ambusher to open-terrain pursuer. The rarity of articulated skeletons leaves gaps in our understanding. Did these species hunt cooperatively, or did each carve out solitary paths through icy woodlands? Feather impressions found in Arctic contemporaries raise the possibility that Antarctic theropods too sported insulating plumage. Although no feathers have been directly preserved alongside Cryolophosaurus bones, its close kin in higher northern latitudes display complex feathers ideal for thermal regulation. If southern theropods inherited similar traits, they may have flicked their plumage to ventilate during long summer days and fluffed it to trap heat during frigid nights. Confirming this requires a fossil of soft tissue or melanosome traces, a discovery paleontologists eagerly anticipate to complete the picture of polar predator biology. Nighttime predation under seasonal darkness poses another puzzle. Arctic theropod trackways show creatures active during periods of extended night, suggesting acute low-light vision or reliance on other senses. Cryolophosaurus possessed large orbits, and its sclerotic rings hint at an eye optimized for dim conditions. Whether it relied on stealthy ambush tactics or pack coordination under twilight remains unknown. Reconstructing its hunting strategies will depend on finding more complete skulls and inner ear structures that reveal balance and acoustic sensitivity. Cold climates impose energetic challenges on active predators, demanding high metabolic rates or behavior adaptations such as basking and torpor. Isotopic studies of oxygen in theropod bones suggest temperature-dependent growth fluctuations, with growth arrests during polar winters. Did these carnivores enter brief hibernation or migrate to lower latitudes? The discovery of bone beds containing both juveniles and adults argues against long-distance migration, implying an ability to withstand months of darkness and cold, a feat requiring yet undocumented physiological innovations. Pathological evidence in theropod fossils offers further insight into their harsh lives. Healed bite marks on ribs and vertebrae indicate intraspecific combat, perhaps over territory or mates, in resource-scarce polar environments. Arthritis in ankle joints of some specimens suggests that individuals survived repeated trauma, possibly from chasing large prey or navigating uneven frozen terrain. These scars, etched in bone, speak of resilience but also raise questions about how wounds were treated or protected from infection in near-freezing temperatures. Trackways of mixed-size theropods in coastal sediment layers point to potential social foraging or family groups. Small tracks flank larger ones in parallel arrays, implying that juveniles may have followed adults during hunts or migrations. This dynamic could have offered young predators protection and hunting lessons, yet it also increased competition for scarce prey. Deciphering whether these patterns represent coordinated packs or coincidental crossing of paths remains a central challenge in understanding polar theropod behavior. The role of volcanic activity in shaping theropod evolution cannot be overlooked. Many fossils lie within ash-rich strata, indicating that these predators endured periodic eruptions and pyroclastic flows. Such events may have driven opportunistic feeding on weakened herbivores and triggered rapid dispersal across ice-free corridors. The interplay between volcanic disturbance and predator-prey dynamics adds a layer of complexity to polar ecosystems, hinting at boom and bust cycles that influence theropod diversity and distribution. Antarctic theropods stand as testaments to the adaptability of predatory dinosaurs, forging lives in extremes of light and cold that few modern creatures could endure. Each bone fragment, footprint, and pathological scar carries secrets of hunting strategies, social interactions, and physiological mastery of polar realms. As new expeditions probe deeper into frozen layers, the Ice Age theropods promise to reveal further mysteries, predators shaped not just by prey, but by the relentless rhythms of sun and snow. Part 3. Cold-blooded giants. The paradox of giant dinosaurs surviving within the polar twilight hinges on their thermoregulatory strategies. Sauropods weighing tens of tons, such as those hinted at by fragmentary limb bones on James Ross Island, could not rely on simple ectothermy, absorbing heat from ambient air. Instead, their massive volumes conferred inertial homothermy, where a low surface area-to-volume ratio slowed heat loss, maintaining relatively stable core temperatures despite frigid night air. Yet this passive mechanism alone seems insufficient for months of darkness, suggesting a more active physiological component, perhaps a moderate metabolic heat production that blurred the line between cold and warm-bloodedness. Bone histology provides crucial clues to metabolic rates. Polar sauropod bones reveal dense fibrolamellar tissue characterized by a woven matrix, and abundant vascular canals, traits associated with rapid sustained growth and high metabolism. Interspersed within these growth fabrics are seasonal lines of arrested growth, marking winter slowdowns. The juxtaposition of fast-growing bone with winteral pause lines indicates that these giants ramped up metabolism during summer abundance, then entered a throttled-down state during months of scarcity. Such dynamic shifts suggest a form of facultative endothermy, active heat generation when resources permitted, combined with economical dormancy. Theropods too exhibited signs of metabolic flexibility. Cryolophosaurus femora show tightly spaced vascular canals, and the absence of extensive haversian remodeling features consistent with high growth rates and elevated metabolic demands. Yet, oxygen isotope ratios from these bones fluctuate seasonally, implying cooler body temperatures in winter months. This cyclical thermal pattern hints at heterothermy, dinosaurs that could upregulate heat production during active seasons and then allow their body temperatures to drop, conserving energy when prey was scarce and darkness reigned. Gigantothermy's effects extended to polar light regimes. During midsummer's continuous daylight, high-latitude dinosaurs likely faced overheating risks. Large sauropods may have sought shade beneath towering araucaria and podocarp canopies, using shallow water bodies to dissipate excess heat, a behavior modeled by modern hippos. Conversely, during polar winter, metabolic heat and group clustering could have provided microclimates within nesting or resting sites, reducing individual energy expenditure. These behavioral adaptations, though invisible in the fossil record, are inferred from the ecological demands of extreme photoperiods. The persistence of small-bodied dinosaurs in polar zones further complicates the metabolic narrative. Troodontids and small ornithomimids, lightweight and feathered, likely relied on high metabolic rates, akin to modern birds, maintaining constant body heat through insulation and active thermogenesis. Their success at the poles suggests a spectrum of thermoregulatory strategies among dinosaurs, from inertial home othermy in giants to true endothermy in smaller taxa, all finely tuned to exploit seasonal windows of resource abundance. Comparisons with modern reptiles offer limited analogies. Large leatherback turtles, for instance, maintain elevated core temperatures via gigantothermy and intermittent flipper movements, while Komodo dragons bask to raise temperatures for digestion, yet neither model fully captures the complexity of dinosaur metabolism. The unique combination of fibrolamellar bone, potential air sac ventilation, and gigantism implies dinosaurs forged novel physiological pathways, metabolic mosaics tailored to their size and latitude. Feathered insulation also emerges as a metabolic tool. Though direct evidence of feathers on Antarctic theropods is lacking, their close relatives in the Arctic possessed complex plumage. If southern dinosaurs shared this trait, feathered theropods and even feathered sections on large sauropods could have reduced convective heat loss. Fossil skin impressions of hadrosaurs and nodosaurs suggest varied integument. Insulated patches might have served to stabilize critical body regions, such as the neck and torso, where heat conservation was most necessary. The debate over dinosaur thermoregulation remains one of paleontology's most profound mysteries. Recent models integrate bone histology, isotopic data, and biomechanical simulations to produce temperature profiles across seasons. Yet without living dinosaurs for direct observation, these reconstructions rely on inference and analogy. Each new polar fossil has the potential to refine or overturn prevailing models, underscoring the tentative nature of our current understanding. Ultimately, the image of cold-blooded giants stumbling through frigid forests gives way to a vision of metabolic hybridity. Polar dinosaurs appear neither fully ectothermic nor truly endothermic, but occupied a middle ground, strategically alternating between heat conservation and active thermogenesis, buoyed by their colossal sizes and physiological innovations. This energetic versatility allowed them to master extremes of light, temperature, and resource availability, carving out a niche at Earth's icy edges that few modern animals could emulate. Part 4. Bone Histology. The microscopic architecture of polar dinosaur bones reveals a hidden chronicle of growth, climate, and physiology. Thin sections of sauropod limb bones from Antarctica display a tapestry of fibrolamellar tissue, woven bone matrix interlaced with dense vascular canals, indicative of rapid sustained growth during the sunlit summer months. Yet, within this exuberant bone fabric lie narrow, dark lines known as lines of arrested growth, lades each marking a seasonal slowdown when daylight dwindled. The spacing and number of lades offer a year-by-year ledger of individual life histories, yet the precise triggers for these growth halts, cold temperature, food scarcity, or hormonal cycles, remain tantalizingly unresolved. Theropod histology paints a complementary portrait. In Cryolophosaurus, cross-sections of the femur show a transition from embryonic woven bone to a more organized lamellar structure as individuals matured. Midshaft regions retain primary osteons arranged in radial patterns, suggesting high blood flow to skeletal tissues. Closer to the periosteal surface, secondary remodeling indicates ongoing bone turnover, a potential sign of mechanical stress adaptation in frigid environments. The balance between bone deposition and resorption hints at dynamic skeletal maintenance, yet how polar predators optimized bone strength without compromising metabolic economy remains an open question. Hadrosaur bone plates preserved on Seymour Island reveal unexpected histological specializations. Their dermal ossifications, once thought inert, show internal networks of vascular channels akin to those in mammalian antlers. Such vascularization could have supported rapid growth or thermoregulatory functions, channeling blood to dissipate or conserve heat. These plate-like structures exhibit growth rings of their own, suggesting that dermal armor expanded in pulses, perhaps tracking seasonal resource availability. The evolutionary drivers of such osteological extravagance in polar hadrosaurs, whether for display, defense, or thermal control, continue to spark debate. Microscopic examination of ankylosaur osteoderms from Antarctic outcrops uncovers remarkable density contrasts. The outer cortex consists of compact bone with minimal porosity, providing formidable protection, while the inner cancellous core displays a honeycomb-like trabecular arrangement optimized for energy absorption. These contrasting architectures point to dual roles: rigid defense against predators and shock mitigation during falls or intraspecific jousting. Yet the degree to which these armored giants tuned osteoderm thickness seasonally or across life stages remains unknown, a mystery locked within fossilized bone tissue. In juvenile polar dinosaurs, growth rings cluster tightly near the medullary cavity, emphasizing accelerated early growth. Histological samples of sauropod embryos and hatchling bones show centralized fibrolamellar deposition that fans outward, constructing the early skeleton with astonishing speed. This rapid ontogenetic burst may have been crucial for survival in harsh climates, allowing young dinosaurs to reach sizes less vulnerable to predators and cold. However, the cellular mechanisms enabling such swift ossification, stem cell proliferation rates, growth factor regulation, remain speculative, awaiting molecular proxies seldom preserved in deep time. Isotopic microanalysis of bone phosphate, layered within histological sections, offers subtle glimpses into paleotemperatures. Variations in oxygen isotope ratios between consecutive growth rings suggest summer warmth of up to 20 degrees Celsius and winter chills nearing freezing, an extreme thermal amplitude that tests the limits of reptilian physiology. Integrating these isotopic patterns with histological markers of metabolic downshifts could unlock the chronology of polar dinosaur life cycles, yet aligning chemical and structural records at subannual scales remains a formidable methodological hurdle. Bone microdamage and repair traces add a biomechanical dimension to the histological narrative. Microcracks in the lamellar bone of sauropod ribs point to repetitive loading, likely from breathing and arboreal browsing motions, while clusters of resorptive pits adjacent to these cracks reveal cellular remodeling activity. Such evidence of lived-in skeletons underscores the resilience of polar giants, yet also prompts questions about bone fatigue thresholds and recovery during prolonged winters when metabolic rates plummeted. The interplay between mechanical stress, microdamage accumulation, and seasonal repair remains largely uncharted territory. Advances in synchrotron imaging now allow three-dimensional histology, unveiling the spatial distribution of vascular canals and osteocyte lacunae without destructively slicing fossils. Early applications to Antarctic dinosaur bones have revealed unexpected branching patterns in blood vessels, possibly linked to localized heat dissipation or nutrient exchange. These volumetric datasets hint at histological complexities far beyond two-dimensional assumptions, but interpreting functional significance from canal geometry presents a fresh suite of enigmas for paleobiologists and engineers alike. The study of dinosaur bone histology in polar contexts weaves together threads of growth, physiology, and environment into a rich but incomplete tapestry. Each histological slide illuminates facets of ancient biology, rapid neonatal growth, seasonal metabolic shifts, structural reinforcement, yet also casts new shadows, revealing gaps in our understanding of cellular processes and environmental interactions. As microscopic techniques advance and more polar specimens come to light, the histological record promises to further unravel the mysteries of how dinosaurs inscribed their lives into bone under the extremes of light, cold, and time. Part 5. Growth rings. Polar dinosaur bones bear annual rings akin to those in ancient trees, each concentric band marking the passage of a year under shifting light and temperature extremes. In sauropod limb bones from James Ross Island, closely spaced rings near the periosteal surface reveal a protracted childhood, followed by dramatically wider rings, signifying adolescent growth spurts, before narrowing again in adulthood. The alternating thick and thin bands trace a biography of resource abundance during endless summer days and metabolic restraint during long, frigid nights. Yet the precise drivers of growth slowdown, whether hunger, cold, or hormonal cues, remain elusive. Theropod growth rings tell a complementary tale. In Cryolophosaurus, femoral cross-sections display double lines of arrested growth, lay-gaze in particularly cold years, hinting at catastrophic winters or volcanic ash fallouts that imposed an extra annual stress. These twin lay-gaze might record two distinct halt events, one from early winter chills and another from a sudden climatic anomaly. Interpreting such anomalies demands correlating bone records with sedimentary evidence of volcanic layers or paleoclimate proxies, a synthesis still unfolding in the polar strata. Hadrosaur bones from Seymour Island reveal intriguing irregularities in ring spacing. While most annual rings cluster regularly, some zones exhibit compressed bands that could indicate brief periods of stunted growth, possibly tied to drought-driven wildfires or sudden shifts in plant communities. Within these compressed zones, microscopic resorption cavities suggest partial remodeling of earlier bone, as if the dinosaur's physiology attempted to reclaim resources from existing tissue during harsh seasons. How these animals balanced survival with skeletal maintenance remains a frontier of polar paleobiology. Smaller ornithopods show a contrasting ring pattern, a rapid early growth phase with minimal lag deposition, implying juveniles pushed hard to reach sizes that conferred predator safety. After this initial burst, growth rings become more pronounced and regularly spaced, reflecting a shift to seasonal growth dictated by resource availability. This ontogenetic switch suggests a life history strategy fine-tuned to polar extremes. Grow fast when energy intake is high, then conserve during the long winter dark. Stress-induced rings, thicker than normal, occasionally punctuate the histological record of both herbivores and carnivores. In ankylosaur osteoderms, these stress rings may correspond to years of high parasite loads or disease outbreaks, forcing the animal to divert energy from growth toward immune responses. The presence of such rings raises questions about disease ecology in ancient polar environments. What pathogens thrived in cold ecosystems? And how did dinosaur immune systems adapt to seasonal pathogen pressures? Comparative studies of polar versus temperate dinosaur fossils uncover stark contrasts in ring patterns. Mid-latitude sauropods often show multiple lags only in response to extreme drought, whereas Antarctic specimens bear lags annually without exception. This suggests that even mild winter stresses in polar habitats were sufficient to halt growth each year. The physiology required to endure these annual growth arrests, rapid metabolic downregulation, and efficient protection against cold tissues, reflects remarkable evolutionary innovation. Isotopic analyses paired with growth rings enable reconstruction of seasonal climates. Oxygen isotope ratios measured in successive rings of a single bone reveal temperature swings of up to 20 degrees Celsius between summer and winter months. When matched to ring widths, these temperature estimates help decode whether growth ring thickness correlates more strongly with temperature or food availability. However, conflicting signals, wide rings in cooler than expected years, indicate that other factors such as plant nutritional value also played crucial roles. Bone growth rings occasionally disappear in the outermost cortex of elderly individuals, forming a terminal growth zone where remodeling eclipses new deposition. This zone offers a glimpse of senescence in dinosaurs, a period when energy resources shifted from growth and repair to reproduction and maintenance. In polar dinosaurs, the timing and duration of this terminal phase remain uncertain, with some giants showing few outer rings before death, suggesting shortened lifespans or abrupt mortality events. Ultimately, growth rings inscribed in polar dinosaur bones serve as natural archives of life under extreme conditions. Each lag and each expansion ring locks in data on environmental stress, metabolic adaptation, and the resilience of these ancient creatures. Still, correlating these signals across species, seasons, and geologic events poses a complex puzzle. As histological methods refine and more specimens come to light, the silent annals of growth rings may yet reveal the full seasonal drama of dinosaurs at Earth's frozen edge. Part 6. Feather insulation. Feathers, once thought exclusive to birds, have emerged as critical adaptations for polar dinosaurs, providing insulation against relentless cold. Fossil discoveries in high-latitude sites, such as the Liaoning province, reveal that even sizable theropods, like UT ranus, bore filamentous feathers capable of trapping heat. Although direct feather impressions remain absent from Antarctic specimens, close relatives of known feathered genera inhabited polar latitudes in the northern hemisphere, suggesting convergent evolution of insulating plumage. The presence of insulation on multi-ton predators and herbivores alike implies that feathers were more than display structures. They were thermal blankets that enabled survival in frigid climes. Among small Maniraptoran theropods, complex pennaceous feathers likely formed dense coats. These feathery arrays, composed of branching barbs and barbules, created trapping pockets of warm air. Polar stations may have accessed these microclimates by tucking heads beneath wing folds or huddling in shaded depressions, while overlapping feather tracts across the torso, limbs and tail provided full body coverage. The energetic cost of growing and maintaining such extensive plumage must have been significant, prompting questions about molt cycles, whether polar dinosaurs synchronized feather replacement with seasonal resource peaks or retained worn feathers through lean winters to avoid metabolic spikes. Evidence from melanosome analysis adds color to the insulation story. Darker pigments absorb more solar radiation, so a predominantly black plumage would have conferred thermoregulatory benefits during low-angle winter sunlight. In Microraptor, melanosomes indicate iridescent black plumage, and while Antarctic taxa lack direct pigment data, similar structures could have evolved to maximize heat uptake. Whether polar dinosaurs selectively bred for darker feathers, trading camouflage for warmth, remains conjectural, yet feather color likely balanced competing needs of concealment, signaling, and thermoregulation. Feather structure itself may have varied seasonally. Modern birds exhibit seasonal plumage changes, growing thicker down layers in autumn before shedding them in spring. Polar dinosaurs could have honed similar strategies, augmenting their insulating cover in anticipation of winter darkness. Feather follicles embedded deep in the skin would have supported rapid growth of downy filaments, while specialized prein glands might have produced oil to waterproof plumage and trap warmth. The fossil record has yet to capture these soft tissue features, rendering their existence a captivating hypothesis awaiting validation. Large-bodied dinosaurs, such as polar hadrosaurs, challenge assumptions about feather coverage limits. While miniaturized feathers suit small thermoregulators, covering a 5-ton hadrosaur in dense plumage would risk overheating during long summer days. Instead, these giants may have possessed localized insulation patches around the neck, flanks, and dorsal regions where heat loss threatened core temperature. Skin impressions from related species reveal scale mosaics interspersed with filamentous structures, possibly proto-feathers, hinting at a hybrid integument that optimized thermal control without suffocating broad backs and flanks. Behavioral adaptations complemented feather insulation. Herds of feathered ornithomimids might have huddled in tight formations, sheltering individuals from biting winds. Thermoregulatory postures, tucking limbs and tails to minimize exposed surface, would have amplified feather effectiveness. Some tracks show parallel prints fading into resting impressions, suggesting that polar dinosaurs paused and crouched in snowdrifts, using feathers to create insulated nests against the frozen ground. Whether such resting behavior left identifiable trace fossils remains an open question for ichnologists. Comparisons with modern analogues, such as ptarmigans and penguins, yield further insights. Arctic ptarmigans grow dense under feathers and shift from brown summer plumage to white winter coats, blending camouflage with insulation. Antarctic dinosaurs may have mirrored this seasonal color change, though paleobotanical evidence of continuous canopy cover might have reduced the necessity for white winter plumage. Penguins' tightly packed feathers and waterproof oils facilitate survival in icy waters. Avian echoes that suggest similar integumentary oils existed in some polar dinosaurs, but the molecular composition of these oils remains a mystery locked in vanished follicles. The evolution of feathers as insulation underscores a broader trend of physiological innovation among polar dinosaurs. Feathers likely interacted with respiratory air sac systems, allowing heat exchange between exhaled air and cooler incoming air, minimizing thermal loss. This integrated warming cycle would have sustained high metabolic rates during active seasons and prevented fatal chilling during rest. Yet direct evidence of such heat exchange structures in fossilized soft tissues eludes discovery, leaving the extent of respiratory dermal collaboration an unsolved riddle of paleobiology. Feather insulation in polar dinosaurs represents a nexus of anatomy, behavior, and environment. An adaptation that transformed dinosaurs from cold, vulnerable reptiles into masters of extreme climates. Each feather follicle, each melanosome trace, and each inferred molt cycle weaves into the grand narrative of survival under solar scarcity and icy winds. As paleontologists uncover more feathered fossils from high latitudes, the tapestry of polar dinosaur life grows ever richer, promising revelations about how feathers first ventured beyond flight and display to cloak giants in warmth. Part 7. Midnight Sun Nests. In the ancient polar regions, dinosaurs did not merely traverse the icy landscapes, they nested and raised their young amidst the extremes of light and darkness. Fossil evidence from the Prince Creek Formation in northern Alaska reveals that dinosaurs, including both large and small-bodied species, reproduced at high latitudes. The discovery of perinatal and very young dinosaur remains indicates that these creatures did not migrate south to lay eggs. Instead, they remained through the seasons, enduring prolonged daylight during the summer months. This raises a host of questions about how these animals adapted to the polar environment, particularly during the critical stages of reproduction. One of the central mysteries revolves around the thermal regulation of nests. In regions where summer days stretch into continuous light and the temperatures, while warmer than winter, are still relatively low, how did dinosaurs incubate their eggs? Did they use vegetation to create compost-like warmth? Did they rely on body heat? Some nesting dinosaurs, like troodontids, are hypothesized to have partially buried their eggs and sat atop them, much like birds. In colder climates, such behavior would have required significant metabolic commitment. The strategies they used may offer clues to their physiology and whether they leaned more toward warm-bloodedness. Evidence of hadrosaur nesting behavior in these northern latitudes further deepens the mystery. Juvenile hadrosaur bones found in the Arctic are not just recently hatched. They suggest that these young dinosaurs lived for months in polar conditions before reaching sizes suitable for migration. This indicates a long nesting and rearing period, pointing to complex parental investment. These dinosaurs likely had to construct nests that could endure environmental extremes and provide consistent conditions for the developing young. The seasonal extremes in light presented another challenge. Polar summers brought constant daylight, while winters cloaked the region in long darkness. For creatures that laid eggs and raised young under these shifting cycles, timing was critical. Nesting too early risked exposure to freezing temperatures, while nesting too late meant hatchlings might not grow enough to survive the winter. How these species synchronized their reproduction with such narrow windows of optimal climate remains a topic of debate. The distribution of nest sites in polar sedimentary deposits suggests a preference for specific terrains. Elevated ridges with good drainage, proximity to water sources, and protection from wind likely influenced where dinosaurs chose to lay eggs. Yet, few intact nests have been discovered in these regions, making it difficult to reconstruct nesting behaviors with certainty. This absence poses a challenge. Were polar nests less likely to fossilize due to their construction or placement, or have they simply not yet been found? Predation was another factor to consider. In modern bird colonies in the Arctic, predators often pose a significant threat to unguarded nests. Dinosaurs, especially smaller theropods or herbivorous species like Pachyrhinosaurus, may have developed group strategies to protect nesting grounds. Fossil evidence shows a surprising density of juvenile bones in certain formations, hinting at possible communal nesting grounds or creches. If such social nesting behavior existed, it would suggest advanced cognitive and social organization. Then there is the question of brooding. Did some polar dinosaurs stay with their nests like modern birds, shielding eggs from the elements and predators? Large-bodied dinosaurs like hadrosaurs may not have brooded in the traditional sense, but might have used their mass to moderate nest temperatures or protect against weather fluctuations. Alternatively, smaller theropods with feathers may have employed more direct brooding, wrapping their bodies around eggs to conserve warmth, in a behavior reminiscent of modern birds. The high-latitude nesting evidence has significant implications for dinosaur metabolism. The ability to reproduce and raise young in such climates suggests a level of metabolic sophistication not traditionally associated with reptiles. Endothermy, or at least a mesothermic condition, may have enabled these dinosaurs to maintain body heat, incubate eggs, and raise hatchlings in cold conditions. These adaptations support the idea of a complex physiological range across different clades of dinosaurs. Despite ongoing discoveries, the details of nesting in the midnight sun remain elusive. With limited fossilized nests and few eggs found in situ, paleontologists are left piecing together behavior from scattered clues, bone growth rings, sediment layering, and rare juvenile specimens. Every new fossil found in these polar regions has the potential to reshape assumptions about dinosaur reproduction in extreme environments, leaving the story of Arctic nesting a puzzle still far from solved. Part 8. Migration Paths. For a long time, it was assumed that dinosaurs in polar regions would have had to migrate to survive. After all, the extremes of light and darkness, especially in places like what is now northern Alaska, seem incompatible with reptilian life. Yet fossil discoveries have begun to rewrite this assumption. In the sediments of the Prince Creek Formation, near the ancient Arctic coastline, remains of hadrosaurs, ceratopsians, and small theropods have been unearthed in surprising abundance. These fossils aren't limited to adult specimens either. Juvenile bones and even embryos show that dinosaurs not only lived in these high latitudes, but nested there. This raises a question that continues to perplex paleontologists. If the Arctic was their home, did they truly migrate, or did they brave the long polar winters in place? Hadrosaurs, often compared to modern-day caribou in their presumed behavior, have become a focal point in the migration debate. Some researchers once imagined herds of these duck-billed dinosaurs trekking thousands of kilometers to escape the freezing months, much like today's large mammal migrations. But there's a snag. There's no direct fossil evidence of such large-scale movement. Migration leaves traces, pathways, mass death assemblages, directional wear on hooves or bones, but these are lacking in the Alaskan fossil record. Instead, there's evidence of year-round residence. Growth rings in hadrosaur bones suggest that these creatures experience seasonal slowdowns, akin to hibernation-like stasis, rather than fleeing the cold entirely. Small theropods like Troodon further complicate the picture. With large eyes and sharp vision, they were well adapted for low-light conditions, an essential trait for surviving the months-long twilight of the polar winter. The presence of juvenile Troodon remains indicates that they too reproduced locally, suggesting they stayed in these frigid regions year-round. The idea of migration seems particularly unlikely for animals so small and likely warm-blooded, given the energetic cost of long-distance travel and the vulnerability of their young during such a journey. Instead, their strategy may have been behavioral, utilizing burrows, altering activity cycles, or exploiting microclimates to survive. But not all species may have been permanent residents. There's speculation that some larger herbivorous species, especially those with strong limb structures and higher mobility, may have undertaken seasonal movements, though not across entire continents. Instead, they may have followed river valleys and coastal plains, shifting only a few hundred kilometers to find better food sources during the darkest months. These partial migrations could have functioned as survival strategies without requiring the epic scale imagined in earlier theories. Fossil eggshell fragments found in polar strata lend further weight to the residency argument. In several cases, the shell pieces belong to species that would have required months-long incubation periods. If these dinosaurs migrated, they would have had to return in spring and stay until late summer to complete the nesting cycle. But the fossil layers suggest uninterrupted occupation, implying that polar dinosaurs were present year-round, at least for key parts of their life cycle. The logistics of migration simply don't add up for such time-bound behaviors. Then there's the climate factor. Though the Mesozoic poles were warmer than today, they still experienced prolonged darkness and seasonal cold, including snowfall in some regions. Yet despite these conditions, flora such as ferns, cycads, and conifers survived, suggesting a marginal but stable food web. Dinosaurs living in these environments would have adapted to cyclical food scarcity, perhaps storing fat, changing diets, or reducing activity. If that's the case, then migration wasn't a necessity, but one of many options on the ecological table. One fascinating clue comes from bone histology. Examining microscopic cross-sections of polar dinosaur bones reveals distinctive growth lines, slowdowns in tissue development, consistent with seasonal stress. These rings, akin to those in trees, show patterns that are consistent with overwintering rather than migration. They represent a physiological response to environmental strain, not the physical strain of long-distance travel. These lines are present in both juveniles and adults, suggesting a shared pattern of surviving the winter together in place. Still, mysteries remain. If these dinosaurs were year-round residents, how did they cope with the scarcity of food and light? No direct evidence of hibernation structures like burrows or nests insulated for warmth has yet been found in large species, nor is it known whether some polar dinosaurs changed their metabolism seasonally, a feat no modern reptile can replicate. The question of whether they had behaviors resembling torpor, huddling, or cooperative sheltering remains open. These possibilities hint at complexities in dinosaur biology still hidden beneath the ice and stone. What is increasingly clear is that the old binary, migrate or die, no longer holds up under scrutiny. Dinosaurs in polar regions adapted in myriad ways, some staying put through months of darkness, others perhaps shifting ranges subtly with the seasons. The fossil record continues to surprise, offering glimpses of ancient creatures that were far more flexible, resourceful, and ecologically nuanced than previously thought. Whether by instinct or inherited memory, their migration paths or lack thereof remain etched in bone, waiting to be fully understood. Part 9. Polar diets. At first glance, the polar regions of the Cretaceous might seem like an unlikely place for large reptiles to survive, let alone thrive. Yet fossil beds in areas like the Prince Creek Formation of Alaska and the Gippsland Basin of Australia reveal evidence of diverse dinosaur communities that lived well within the Arctic and Antarctic circles. The question of what these creatures ate, especially during months of freezing darkness, has intrigued paleontologists for decades. Unlike modern herbivores that rely on evergreen foliage or store food, dinosaurs had to find ways to endure long stretches of minimal vegetation. That they did so successfully, even raising offspring in such environments, points to complex and fascinating dietary strategies that evolved in isolation over millions of years. Hadrosaurs, often dubbed the cows of the Cretaceous, were widespread in polar fossil records, and their dental batteries, a complex system of hundreds of tightly packed teeth, allowed them to grind tough plant material efficiently. In polar latitudes, summer months provided a brief window of explosive plant growth, including conifers, horsetails, and ferns, which these herbivores could feast on. But it's the long, lean months of darkness that raise questions. Some theories suggest that hadrosaurs may have had browsing techniques similar to modern moose, foraging on twigs and bark in winter. Their beak-like mouths and strong jaw muscles hint at the ability to strip bark from trees, behavior typically not associated with reptiles, but perhaps crucial for survival in such a place. Ceratopsians like Pachyrhinosaurus also populated the ancient polar forests. These beaked herbivores may have depended on the low-lying understory plants, including ground-level ferns and shrubs, that could have remained somewhat accessible even through snow cover. The presence of juveniles in fossil beds implies they stayed in the region year-round rather than migrating, and so their diet had to be consistent with year-round availability. It's possible they employed a root and bulb feeding strategy in the winter, digging for carbohydrate-rich tubers using their powerful forelimbs and sharp beaks. Their short, broad faces and strong biting force make this a plausible feeding adaptation. Theropods, such as the small but intelligent Troodon, present an entirely different set of dietary questions. With teeth suited for slicing flesh and large eyes capable of enhanced night vision, Troodon is thought to have had an omnivorous diet, possibly consuming small vertebrates, carrion, and even vegetation or fungi when prey was scarce. Fossilized coprolites associated with similar species contain bone fragments and traces of plant matter, adding to the picture of an opportunistic feeder. This flexibility would have been a significant survival trait in a region where prey animals likely hibernated, migrated, or were simply less active during prolonged winters. Another intriguing predator, Nanuqsaurus, the polar tyrant, was a smaller cousin of Tyrannosaurus rex, adapted for life in the cold north. While many details of its lifestyle remain speculative due to fragmentary remains, its presence in high-latitude fossil beds hints at permanent residency. If it hunted year-round, its prey may have included juvenile hadrosaurs or smaller dinosaurs like Troodon, as well as reptiles or mammals active in winter. However, scavenging likely played a major role in its diet, with heightened olfactory senses helping it locate frozen carcasses buried beneath snow or ice. The metabolic needs of such a predator in a resource-scarce environment remain a mystery, raising questions about seasonal torpor or slowed metabolic states. Plants in the Cretaceous polar regions exhibited their own seasonal patterns, with rapid growth during long sunlit summers, followed by months of dormancy. Fossilized leaves show adaptations to extreme photoperiods, such as rapid photosynthesis and early leaf drop. For herbivores, this created a narrow seasonal window to feed and bulk up. Some species may have gorged during summer and stored fat, like modern Arctic animals do. Others might have relied on gut fermentation and extended digestion times to extract nutrients more efficiently during lean months. A trait hinted at by large gut cavities seen in some fossil skeletons. Interestingly, some fossilized remains contain evidence of gastroliths, stones swallowed to aid digestion. This behavior would have helped grind tough, fibrous plant matter, especially when softer vegetation was unavailable. Gastroliths are typically found in association with sauropods, but some polar ornithopods may have used similar strategies. These internal grinding mills would have provided a digestive edge in processing hardy frozen plant tissues that other animals could not digest effectively. Evidence from microfossils such as pollen and spores found alongside dinosaur remains further supports the presence of a wide variety of seasonal food sources. These tiny fossils paint a picture of lush summer forests rich in deciduous plants, which would have dropped their leaves just as dinosaurs entered the long winter. Fossils of mushrooms, liverworts, and mosses also appear, hinting that some dinosaurs may have supplemented their diet with fungi, an unusual but high-nutrient food source available even during colder seasons. This ecological flexibility may have been a key factor in the persistence of dinosaur populations in extreme environments. The question of polar dinosaur diets is ultimately one of survival strategy in an unpredictable world. Whether they stayed put, migrated, or employed unusual feeding habits, these species succeeded where few other reptiles dared to tread. Fossils continue to provide tantalizing clues, but much remains uncertain. Did some of them slow their metabolisms like modern reptiles? Did they exploit unique food sources not yet preserved in the fossil record? These mysteries remain open, frozen in time beneath layers of sediment and ice. Part 10. Permafrost tissues. Exceptional preservation in permafrost has unveiled soft tissue remnants that once seemed impossible to recover from dinosaur fossils. In northern Alaska's frozen ground, rare finds of mummified hadrosaurs reveal impressions of skin and hints of muscle fibers, preserved by rapid burial in sediment, followed by freezing temperatures. These cold, stable conditions inhibit bacterial decay, allowing delicate tissues once thought long gone to linger for tens of millions of years. Such discoveries transform our understanding of dinosaur biology, offering snapshots of external morphology and internal anatomy far beyond what bones alone can provide. One of the most celebrated specimens is a partial mummified hadrosaur, whose skin reveals a mosaic of scales and possible keratinous structures. The dorsal skin impressions show hexagonal and polygonal scutes, their relief preserved in 3 dimensions. Under microscopic examination, collagen-like fibers appear as faint, fibrous strands within the skin matrix, hinting at tensile strength and elasticity. These fibers, likely part of the dermal connective tissues, raise questions about the mechanical properties of dinosaur skin, how it stretched over muscle and joints, and how it weathered the extremes of polar climate without cracking or desiccating. Muscle tissue preservation in permafrost is even more astonishing. In some mummified specimens, remnants of muscle bundles and tendon attachments remain visible, suggesting that original soft tissue proteins can survive under the right conditions. Analysis of these tissues shows cell-like structures, though no intact cells per se, implying that cellular membranes have long since degraded. Still, impressions of muscle architecture, fiber orientation and cross-sectional shape provide unique insights into locomotion mechanics. For example, the orientation of preserved thigh muscle fibers hints at the primary direction of force generation during walking, illuminating how these giants moved across frozen plains. Blood vessel impressions have also been reported in exceptionally preserved fossils. Tiny vascular canals within skin and muscle tissues appear as branching networks, once carrying blood to nourish cells and regulate temperature. Their presence raises the possibility that permafrost specimens could yield remnants of hemoglobin or other blood proteins, although chemical analyses have yet to confirm such molecules definitively. If identified, ancient blood proteins could revolutionize our understanding of dinosaur metabolism, oxygen transport, and even immune function. Nervous tissue, however, remains elusive. Despite some claims of neuronal impressions in permafrost-preserved specimens, these reports are contested. Nerve fibers and brain tissues degrade rapidly post-mortem, making their survival highly unlikely. Yet the quest to find any trace of the nervous system continues to drive novel imaging techniques. High-resolution scanning of mummified skulls occasionally reveals impressions in sediment that may correspond to brain case soft tissues, but distinguishing these from taphonomic artifacts demands extreme caution and rigorous verification. Permafrost's role extends beyond individual specimens. In Siberia, crevasse fillings have trapped and frozen Pleistocene mammalian remains alongside plant matter, offering analogues for how dinosaur soft tissues might have fossilized. By studying these more recent permafrost entombments, scientists gain insights into the rates and processes of tissue mineralization and degradation. Extrapolating these findings to Mesozoic permafrost environments suggests that rapid freezing, minimal thaw cycles, and anoxic microzones are key to preserving original biomolecules. Despite its promise, permafrost preservation is not a panacea. Freeze-thaw cycles, even minor ones, can disrupt soft tissue integrity. As climate change accelerates permafrost melt, previously frozen fossils risk degradation before they can be studied. This urgency has prompted the development of remote sensing and rapid excavation strategies to recover specimens before they thaw. The race against time adds a modern twist to paleontological discovery, as researchers scramble to secure permafrost treasures under threat. The potential for permafrost-preserved fossils to yield ancient DNA remains a subject of speculation. While DNA fragments have been extracted from Pleistocene organisms buried for hundreds of thousands of years, the survival of DNA over tens of millions of years is far less certain. Nevertheless, even the recovery of protein fragments, collagen peptides, keratin residues, or blood proteins promises to illuminate evolutionary relationships and physiological traits. These biomolecules could help place polar dinosaurs within the broader dinosaur family tree with greater precision. Permafrost tissues thus stand at the frontier of paleobiological research. Each mummified specimen offers a rare glimpse beyond bones into the living flesh of ancient giants. Yet the mysteries of how, why, and under what precise conditions these tissues endure remain. As new permafrost discoveries emerge and analytical techniques advance, the frozen pages of prehistory will continue to yield startling revelations about the creatures that once roamed Earth's coldest realms. Part 11. Oxygen isotopes. Oxygen isotopes preserved in polar dinosaur bones and teeth have become powerful proxies for reconstructing ancient climates and physiology. By measuring the ratio of heavy to light oxygen isotopes in phosphate molecules, researchers can approximate the water temperatures in which these animals lived and drank. Analyses of hadrosaur bones from the Prince Creek Formation reveal seasonal swings of up to 20 degrees Celsius between summer and winter drinking water, matching the extreme light cycles of the Arctic. The alignment of isotopic cycles with growth rings underscores the intimate link between physiology and environment in these polar giants. In theropod tooth enamel, oxygen isotope ratios provide a high-resolution view of dietary water sources. Troodon teeth show consistent isotopic values through early growth, suggesting hatchlings drank primarily local stream water. As juveniles matured, slight shifts in isotope ratios hint at foraging across broader territories, perhaps tracking melting snowmelt or coastal lagoons during thaw. These subtle variations in tooth chemistry paint a dynamic picture of resource use and movement, revealing how individual dinosaurs navigated changing water networks in a seasonal landscape. Comparisons between species elucidate ecological partitioning. Polar hadrosaurs exhibit a distinct isotopic signature from coexisting ceratopsians, implying that despite overlapping ranges, they exploited different water sources, perhaps preferring riverine versus swampy habitats. This partitioning would have reduced competition in nutrient-poor environments and allowed multiple herbivore species to coexist. The mystery deepens when isotopic data suggest occasional overlap, hinting at rare congregation events, perhaps during mass migrations to verdant feeding grounds or during periods of environmental stress. Oxygen isotope analysis of sauropod vertebrae from Antarctic sites reveals evidence of migratory behavior, or the lack thereof. Some vertebrae show uniform isotopic values, indicating long-term residency in a single climatic zone. Others display marked shifts mid-life, suggesting these giants undertook seasonal movements, likely following meltwater pulses or ephemeral floodplains. The juxtaposition of resident and mobile life histories within the same region underscores the diversity of adaptive strategies among polar dinosaurs. The precision of isotopic thermometry extends to paleo temperatures. In some permafrost-preserved specimens, oxygen isotopes in both bone and surrounding ice layers offer dual records of dinosaur body temperature versus ambient ground temperature. These paired measurements hint at the degree of metabolic heat generation, how much warmer dinosaurs kept themselves relative to freezing soils. Early results suggest moderate endothermy in polar theropods, maintaining body temperatures 5–10 degrees Celsius above their environment, yet allowing partial thermal conformity during resource-scarce winters. Challenges in interpreting oxygen isotope data remain. Diagenetic alteration, post-burial chemical change, can skew ratios, necessitating rigorous screening of samples for preservation quality. Some polar fossils, subjected to freeze-thaw cycles, show subtle recrystallization that complicates readings. Distinguishing primary biological signals from geological overprints is an ongoing endeavor, requiring the development of novel geochemical markers and cross-validation with other proxies like clumped isotopes. Clumped isotope paleo-thermometry adds another dimension, measuring the bonding of heavy isotopes within carbonate minerals to infer formation temperatures independent of water composition. Applied to dinosaur eggshells from mid-latitude sites, these methods have yielded incubation temperature estimates. Extrapolating to polar eggshell fragments hints at incubation in the range of 30–35 degrees Celsius, offering tantalizing clues to nesting strategies under cool summers. Whether polar dinosaurs adjusted nest depth or selected geothermal microhabitats to regulate incubation heat remains a captivating question. Integration of oxygen isotope data with sedimentary records reveals links between dinosaur physiology and paleo-environmental events. Episodes of volcanic activity, recorded by sulfate layers and ash beds, correlate with abrupt shifts in isotopic values, indicating rapid cooling or acid rain effects on water sources. Such correlations suggest that polar dinosaurs not only tracked seasonal cycles, but also responded to cataclysmic events, adapting their water intake and habitat use to survive transient climate shocks. Ultimately, oxygen isotopes unlock a chemical diary of polar dinosaur life, documenting where they drank, how they migrated, and how their bodies coped with extreme seasons. Each isotopic curve, each thermometric reading, deepens the portrait of these ancient inhabitants of Earth's icy fringes. As analytical techniques refine and new specimens emerge, the geochemical whispers preserved in their bones will continue to illuminate the hidden rhythms of dinosaurs beneath the polar sun and shadow. Part 12. Arctic Pathologies. The bones of polar dinosaurs bear silent testimony to the physical hardships of high-latitude life. In the Prince Creek Formation, a Pachyrhinosaurus specimen exhibits fused vertebrae in its neck, a condition suggesting traumatic injury, perhaps from a fall on icy ground or a violent clash with a rival. The subsequent bone remodeling indicates the animal survived long enough for partial healing, revealing resilience in a harsh environment. Such pathologies offer rare windows into personal histories of pain, recovery, and survival under extreme conditions. Theropods like Troodon also display evidence of battle scars. One tibia shows signs of osteomyelitis, a bone infection that likely developed after a deep wound, possibly from prey defense or intraspecific combat. The resulting inflammation and bone overgrowth would have limited mobility, yet the healing pattern suggests the dinosaur lived weeks or months despite a severe handicap. Investigating how polar dinosaurs coped with infection and immobility, did they rely on group support, reduced activity, or elevated immune responses, remains an intriguing puzzle. Fractures in hadrosaur pedal bones hint at the challenges of traversing frozen terrain. Slipping on ice, especially during seasonal thaw, could have resulted in high-energy fractures that in turn bear callus formations indicating healing. These healing rates, inferred from bone histology, appear slower than in comparable temperate specimens, suggesting that colder metabolism or nutrient scarcity in winter months may have prolonged recovery times. Understanding seasonal impacts on healing cycles deepens our appreciation of polar dinosaur physiology. Bone tumors, though exceedingly rare, have been identified in Arctic specimens. A hadrosaur femur shows an exostotic growth, an abnormal bony protrusion that may have arisen from chronic irritation or viral infection. The presence of such lesions raises questions about viral vectors or environmental carcinogens in the Cretaceous Arctic, perhaps linked to volcanic aerosols or unique microbial communities. Untangling the origins of these growths could illuminate interactions between pathogens, environment, and dinosaur immune systems. Dental pathologies provide another layer of insight, where facets and cracks in Troodon teeth suggest episodes of dietary stress, such as grinding against frozen or gritty food sources. Some teeth exhibit reparative dentine, a sign of attempted healing, indicating that theropods could repair minor dental damage. This capacity implies sophisticated physiological processes and perhaps dietary flexibility to compensate for impaired chewing during recovery. Stress indicators in bone microstructure manifest as lines of arrested growth, lags but more subtle stress rings, wider and denser bands, reflect periods of extreme hardship. In ceratopsian scapulae, these stress rings correspond to sediment layers rich in volcanic ash, suggesting rapid environmental upheaval taxed dinosaur health. Whether such stress responses influenced mortality rates during cataclysmic events or merely left biological scars in survivors remains a compelling enigma. Pachyostosis, abnormally thickened bone, has been observed in some polar ornithopod ribs. This condition may have provided structural reinforcement against cold-induced brittleness or buffered against pressure changes during deep dives in icy water. If these dinosaurs fed in shallow marine or riverine environments, as swimming trackways imply, enhanced bone density could serve multiple functions: ballast for buoyancy control and protection against brittle fractures in cold water. Joint pathologies, such as osteoarthritis, appear in aging individuals of multiple species. Hibernation, polished bone surfaces from cartilage loss, marks chronic stress on joints, potentially from walking long distances over uneven tundra or from herding behavior on slippery ground. The prevalence of these conditions suggests that even dinosaurs were not immune to the wear and tear of polar life, and that some individuals reached advanced age before succumbing to degenerative disease. Together, Arctic pathologies paint a vivid portrait of struggle and adaptation. From fractures and infections to tumors and arthritis, these ancient creatures bore the marks of their environment in their bones. Each pathology offers clues to daily life, hazards faced, and the remarkable capacity for survival. As new fossil finds emerge, the catalogue of diseases and injuries continues to grow, enriching our understanding of how dinosaurs lived and often thrived within Earth's frozen extremes. Part 13. Frozen bone beds. Frozen bone beds in polar regions offer captivating snapshots of ancient ecosystems locked in time, where multiple dinosaur skeletons lie jumbled together beneath layers of permafrost and sediment. One such deposit in northern Alaska preserves dozens of individuals, hadrosaurs, ceratopsians and small theropods, intermixed in an odd jumble of articulated and disarticulated remains. The scene suggests a sudden event, perhaps a flash flood over ice-choked rivers that swept carcasses together and buried them rapidly. The quick burial in cold conditions not only halted decay, but also facilitated exceptional preservation, freezing the final moments of these creatures in chilling detail. In Antarctica's Seymour Island, bone beds of small ornithopods and theropods lie beneath volcanic ash layers from nearby eruptions. The juxtaposition of ash and permafrost indicates that these dinosaurs endured alternating threats, choking volcanic debris and grueling cold. The ash likely killed vegetation and poisoned water sources, forcing dinosaurs into marginal areas where they became trapped in icy mud. As the ash settled and temperatures plunged, their remains were sealed in an inhospitable tomb, creating bone beds that combine evidence of volcanic disaster with polar preservation. The Great White North's discovery of a mixed bone bed containing juvenile and adult hadrosaurs raises questions about herd behavior during catastrophic events. Juveniles clustered together, while adults appear peripheral, suggesting a possible protective circle that ultimately failed. The distribution of bones indicates that some animals died standing, their legs splayed in mud, while others lay prone, hinting at rapid entombment in fluid sediments. Deciphering whether this grouping resulted from migration, nesting, or forced congregation at scarce water holes remains a tantalizing challenge. Bone beds of theropods in permafrost reveal another layer of mystery, the presence of multiple species in the same deposit. Small troodontids appear alongside larger dromaeosaurids, their bones mixed in a single stratum. Such associations hint at complex predator interactions. Were these dinosaurs hunting cooperatively, scavenging the same kill sites, or simply drawn together by the same environmental crisis? The absence of articulated skeletons with clear predatory-prey positioning leaves the dynamics of these multi-species assemblages open to interpretation. Micromorphological analysis of bone bed sediments shows finely laminated silt interspersed with organic-rich layers. These alternating strata suggest seasonal freeze-thaw cycles that generated varves, annual layers of sediment deposition. In some bone beds, multiple varves overlying a fossil layer imply that remains remained partially exposed through several seasons before final burial, subject to scavenging and weathering. The survival of fragile bones through these cycles indicates rapid refreezing each winter, with thin insulating sediment layers protecting remains from destructive freeze-thaw damage. Stratigraphic correlation between distant polar bone beds highlights synchronous events. Identical ash layers found across hundreds of kilometers suggest large-scale volcanic eruptions that blanketed vast territories, triggering mass mortality. When coupled with evidence of glacial advance, these layers point to periods where dinosaurs faced the dual pressures of toxic ash and encroaching ice. Bone beds formed in such intervals thus record the intersection of volcanic and glacial forces, two prime movers of polar cataclysms. In some Antarctic bone beds, articulated skeletons emerge intact, their limbs locked in life positions. These specimens, often coupled with skin impressions, speak to the rarest form of preservation, rapid burial under cold, anoxic conditions that halted both bacterial decay and scavenging. The chemical composition of the surrounding permafrost preserves soft tissue morphology, offering glimpses of muscle outlines, skin texture, and even potential traces of melanosomes. Yet these frozen treasures remain few, and each new find reshapes our understanding of dinosaur anatomy and ecology. Bone beds also preserve behavioral traces in the form of trackways and nesting sites interwoven with skeletal remains. Tracks leading away from mass death assemblages suggest survivors fleeing disaster only to meet their own demise. Nest scrapes half filled with carcass fragments point to opportunistic feeding by adults near their nesting grounds. These intertwined behavioral and taxonomic records offer a cinematic view of tragedy and survival rolled into a single fossil layer, a rare glimpse at dinosaurs in motion and at rest. Ultimately, frozen bone beds in polar realms stand as monumental records of both ecological resilience and vulnerability. They reveal how dinosaurs congregated, interacted, and met their end under the relentless forces of nature. Each bed, whether a scattering of scattered bones or a tableau of articulated mummified remains, adds a chapter to the epic of survival amid ice and ash. As ongoing expeditions uncover new bone beds and refined dating techniques link them to global events, the frozen stories of polar dinosaurs continue to unfold, each layer a testament to life on the brink. Part 14. Polar versus Tropic. The polar dinosaurs of the Cretaceous diverged in striking ways from their tropical counterparts, reflecting adaptations to extreme seasonality and cooler climates. In tropical regions, lush vegetation supported large herds of herbivores year-round, while polar ecosystems boasted condensed food webs centered on brief bursts of plant growth. Limb-bone comparisons reveal that polar hadrosaurs possessed relatively shorter, more robust femora, an adaptation likely enhancing stability on uneven, ice-scoured terrain, whereas tropical species display longer, gracile limbs optimized for speed. These morphological differences underscore how environment shapes form, even within closely related clades. Tropical theropods, such as Allosaurus, hunted in open woodlands under consistent day lengths, relying on vision and heat-driven endurance. Polar theropods like Cryolophosaurus, by contrast, evolved larger orbits and wider sclerotic rings, an ocular specialization for dim light. Their semicircular canal structures hint at enhanced balance control, aiding locomotion on slick or uneven surfaces. While both groups shared basic carnivorous anatomies, the polar forms exhibit neural and sensory refinements that reflect the low-light, high-cold challenges of their homes. Skin and integument records further accentuate polar-tropical contrasts. Tropical nodosaurs show uniform armor plating, likely optimized for defense against abundant large predators. Polar nodosaurs appear to have integrated filamentous structures among their scutes, primitive feathers or bristles, providing added insulation. This dual integument mix suggests that polar dinosaurs blended armor and warmth, a compromise unseen in their tropical relatives, whose consistent warmth negated the need for feather-like insulation. Growth patterns offer another window into environmental influence. Tropical sauropods often lack clear lines of arrested growth in their bones, growing almost continuously in favorable climates. In polar sauropods, pronounced growth rings record annual slowdowns, reflecting metabolic throttling during long, cold winters. These cyclical growth patterns not only indicate seasonal adaptability, but also imply hormonal mechanisms for suspending and resuming growth, a biological relay between dormancy and rapid development, absent in equatorial species. Reproductive strategies diverged along latitudinal lines as well. Tropical dinosaurs likely timed breeding to resource peaks, but were not constrained by month-long darkness. Polar species, confronted by narrow breeding seasons, show evidence of highly synchronized nesting, judging from egg clutch uniformity and simultaneous juvenile age classes, ensuring hatchling emergence during maximal daylight. Tropical dinosaurs display wider nesting windows and less clutch synchronicity. The polar insistence on tight reproductive calendars hints at strong selective pressure to align life cycles with environmental rhythms. Dietary isotopes illustrate differences in water and plant source use. Tropical herbivores' bone isotopes reflect stable, warm-season water consumption, while polar dinosaurs exhibit wide isotopic swings tied to melting snow and spring water pulses. Tropical predators' isotopes reveal localized prey cycles, whereas polar theropods show evidence of shifting prey use, fish and small vertebrates in summer, supplemented by scavenged carcasses in winter. These chemical signatures paint dynamic seasonal diets for polar species versus more constant tropical feeding patterns. Social behavior may also split along climate lines. Tropically, herd sizes could swell year-round, with abundant food minimizing competition. Polar herds, facing seasonal scarcity, remain smaller and potentially nomadic, banding together only during brief abundance. Fossil trackway density supports this. Tropical bone beds show vast congregations, while polar bone beds reveal more modest, age-segregated groupings. These social strategies reflect the balance between safety in numbers and resource limitations endemic to high latitudes. The more regulatory anatomy further distinguishes polar from tropical dinosaurs. Tropical species could afford less insulation. Their skin impressions lack evidence of extensive feathers. Polar dinosaurs display thicker down-like coverings, preserved in mid-latitude analogues, and bone histology suggests air sac expansions, enhancing respiratory heat retention. This physiological divergence indicates an evolutionary trajectory where insulation and internal heat recycling became essential for polar survival, but remained niche improvements elsewhere. Ultimately, the polar-tropic comparison reveals the remarkable plasticity of dinosaur physiology and behavior. From limb proportions and sensory adaptations to growth rhythms and social structures, high-latitude species carved out a suite of innovations to conquer extremes. Tropical dinosaurs, basking in equatorial stability, evolved along a different path, one of continuous growth, open environment hunting, and less metabolic fluctuation. These contrasts underscore how geography and climate drive the tapestry of life, weaving distinct yet equally successful strategies across the Mesozoic world. Part 15. Sexual dimorphism. Polar dinosaurs may have exhibited subtle yet significant differences between males and females, though the harsh conditions obscure the fossil record. In large hadrosaurs inhabiting high latitudes, some individuals display broader, more robust skull crests, while others bear slimmer, elongated forms. These variations could reflect male display structures used to attract mates during the brief summer breeding season, or they might indicate differences in feeding strategies between the sexes. The crest's internal passages, once thought solely for vocal resonance, may also have amplified displays of social status, hinting at a world where dimorphic traits served both attraction and dominance. Ceratopsians like Pachyrhinosaurus show intriguing possibilities for sexual dimorphism. While both sexes sported solid nasal bosses, a subset of specimens possesses more pronounced ornamental frill flanges, bony projections that could have been brightly colored in life. These flanges might represent male ornamentation, used in combat rituals or courtship displays beneath the midnight sun. Yet without soft tissue evidence or direct correlation to nesting sites, assigning these features to one sex remains speculative, a frustrating gap in understanding the social lives of polar horned dinosaurs. Theropods offer another window into dimorphism. In Troodon, two morphs of femoral robustness have been identified, gracile bones with slimmer shafts, and more robust ones bearing thicker cortical walls. If these differences represent sex, they could indicate that one sex, perhaps males, required greater locomotor endurance for territorial patrols or mate chasing, while the other favored agility. Yet growth ring analyses suggest both morphs matured at similar rates, complicating interpretations of sex versus ontogeny. The tantalizing overlap of size and growth data leaves the true nature of these morphs an open question. Crested sauropods, such as Cryolophosaurus, hint at sexual display in their transverse head crests. Variations in crest curvature and thickness among specimens suggest individual differences beyond simple ontogenetic change. If these differences correlate to sex, they may have served as visual signals during the polar summer's constant daylight. However, the lack of clearly differentiated juvenile and adult crest development muddy attempts to map crest variation onto sex, leaving the purpose of these crests shrouded in mystery. Trackway studies provide indirect evidence of dimorphism through gait analysis. In some polar track sites, two-size clusters of similarly spaced footprints suggest mixed sex groups moving together, with larger prints potentially indicating males. The presence of parallel smaller prints alongside larger ones hints at protective escort behavior, perhaps males guarding females during nesting migrations. Yet without skeletal remains directly linked to these tracks, attributing them to male, female pairs remains conjectural, a compelling but unresolved detective story, written in stone. Nesting fossils yield further puzzles. At certain polar nesting grounds, clusters of eggs and hatchlings appear associated with adult specimens, showing differing pelvic girdle structures. Some adults exhibit wider pelvic inlets, possibly females adapted for egg-laying, while others have narrower hips, potentially belonging to males. This pelvic dimorphism could mirror modern bird patterns, where female pelvises widen for reproduction. Unfortunately, the fragmentary nature of pelvic fossils prevents definitive sex assignments, leaving this potential clue to polar dinosaur reproduction tantalizingly out of reach. Bone microstructure can sometimes reveal sex-based physiological differences. In modern crocodilians, steroid hormone deposits subtly alter bone growth patterns. Similar chemical signatures might exist in polar dinosaurs, detectable through advanced geochemical analyses of growth bands. Preliminary work shows slight variations in lag spacing and bone density among presumed male and female-sized specimens, suggesting differential energy allocation. Yet isolating hormone-driven signals from environmental stresses remains a formidable challenge, one that combines paleohistology and geochemistry in pursuit of ancient endocrinology. The question of coloration plays a vital role in sexual dimorphism. Melanosome analyses in mid-latitude feathered dinosaurs reveal sexually dichromatic plumage, with males bearing iridescent feathers for courtship. If polar dinosaurs shared this trait, males may have sported darker, more radiant feather patches to attract mates under the summer's continuous light, while females remained cryptically colored for nest camouflage. Direct evidence of melanosomes in polar feathers has yet to surface, but the possibility frames an evocative picture of display behaviors in frigid landscapes. Understanding sexual dimorphism in polar dinosaurs remains one of paleontology's most alluring enigmas. The fragmentary clues, crest shape, bone robustness, trackway proportions, pelvic differences, and potential hormonal signals hint at complex social and reproductive behaviors. Yet without a Rosetta Stone fossil linking dimorphic traits to clear sex indicators, the true nature of male and female dinosaurs in Earth's frozen extremes continues to elude us. Future discoveries, especially of articulated skeletons preserving soft tissue contrasts, promise to shed light on these mysteries of ancient polar love and rivalry. Part 16. Faunal Origins. Dinosaurs of the polar regions traced their lineage through shifting continental connections that allowed dispersal into high latitudes long before the polar ice caps formed. In the early Jurassic, Antarctica lay at mid-latitudes connected to southern Gondwana, permitting genera like Cryolophosaurus to evolve in temperate climates before the land drifted toward the South Pole. As Gondwana fragmented, land bridges and archipelagos provided fleeting corridors for migration, planting theropods, sauropods and ornithischians into emerging polar habitats. Each breakup and reconnection reshaped faunal composition, seeding polar ecosystems with waves of newcomers followed by periods of isolation. By the Cretaceous, a northern arc connected Asia to North America via the Bering Land Bridge, enabling genera such as hadrosaurs and ceratopsians to cross into Arctic Alaska. Fossil parallels between Alaskan hadrosaurs and their Asian cousins reveal astonishing similarities in skull and dental traits, suggesting recent common ancestry. These migrations likely followed flowering plant blooms and insect emergences, tracking summer vegetation pulses. Yet the need to endure polar winters on either end of the journey required rapid physiological adaptation, turning migratory pioneers into permanent residents within a few generations. Antarctica's late Cretaceous faunas show unexpected ties to South America. Titanosaurs discovered in James Ross Island exhibit limb bone proportions akin to South American saltosaurids, hinting at Gondwana and dispersal routes that persisted even as South America began to separate. Ocean currents, island stepping stones and shallow coastal shelves may have facilitated these epic voyages of titanic herbivores. The persistence of these lineages in Antarctica until the end of the Cretaceous underscores how continental drift and sea level changes sculpted a unique high-latitude biota. Australia's polar fossils further complicate the picture. During the mid-Cretaceous, Australia clung to Antarctica, forming a continuous high-latitude corridor. Ornithopods and early ankylosaurs in southeastern Australia share ancestry with Antarctic forms, suggesting faunal interchange across what would become the Southern Ocean. As Australia drifted northward, its polar dinosaurs evolved in relative isolation, giving rise to endemic forms adapted to cooler climates. Yet relic populations of their Antarctic kin may have survived as climates warmed, only to vanish in the end Cretaceous extinction. Europe's high-latitude records add another layer. Although less intensely studied, Cretaceous sites in Russia's Far East and Northern Europe yield spinosaurid fragments and small coelurosaurs alike, aligning with Asian assemblages. These finds hint at a circumpolar fauna encircling the Arctic Ocean, connected by contiguous landmasses before the opening of the North Atlantic. The eventual submergence of these land bridges shut down dispersal pathways, trapping populations in evolving polar pellies that diverged from their lower latitude relatives. Isolation of polar faunas led to intriguing endemic radiations. In Arctic Alaska, small theropods evolved lightweight bills and large eyes suited to dim light, traits not as pronounced in their southern counterparts. Antarctic ornithopods, on the other hand, developed shorter limbs and stockier frames, perhaps as energy-saving adaptations to cooler climes. These divergent morphologies attest to evolutionary experiments conducted in parallel at Earth's extremes, producing forms that, while rooted in shared ancestry, took on novel life histories. Paleobotanical evidence illuminates the backdrop for these faunal dramas. Fossilized pollen and leaf impressions show that polar forests of podocarps, araucarias and ferns provided continuous green corridors during warm intervals, sustaining migrating herds and cloaking land bridges. As climates cooled, these refugia shrank, isolating dinosaur populations on mountain-lined valleys and coastal plains. The contraction and expansion of these vegetated belts orchestrated pulses of dispersal and retreat, weaving the complex tapestry of polar dinosaur origins. The timing of these migrations intersects with global sea level changes. Transgressive regressive cycles exposed continental shelves, opening lowland migration routes, then submerged them, imposing genetic bottlenecks. Each high sea stand fostered isolation, driving local adaptation, while low stands enabled influxes of competing species. This ebb and flow of connectivity and separation generated the mosaic of polar faunas, blending cosmopolitan colonists with endemic holdovers and extinct experiments. Unraveling the origins of polar dinosaurs thus demands a synthesis of plate tectonics, paleoclimate, and phylogenetic analysis. Each fossil discovery refines the story, whether it's a titanosaur femur hinting at Gondwanan ties or a troodontid tooth echoing Asian lineages. Yet gaps remain, key intervals lack fossils, and some migration routes are known only through fragmentary clues. As fieldwork extends into previously inaccessible polar outcrops and molecular techniques sharpen evolutionary timelines, The story of faunal origins at Earth's poles will continue to evolve, revealing how the mighty dinosaurs first ventured into and ultimately mastered the fringes of their world. Part 17. Ancient Flora. Under the midnight sun, polar forests of the Cretaceous flourished in surprising diversity, forming verdant canopies that sustained herbivorous dinosaurs through long summers. Fossilized wood from Antarctica and Arctic sites preserves growth rings that mirror those in dinosaur bones, wide summer bands of rapid ring-porous growth, interspersed with narrow winter bands of slower formation. These rings reveal that trees like araucarians and podocarps thrive during continuous daylight, while surviving lean months of darkness by entering a quasi-dormant state. The rhythmic pulse of growth and arrest in both flora and fauna underscores a shared reliance on seasonal light extremes. Ferns and horsetails carpeted the forest floor, their fronds adapted to low-light photosynthesis. Some frond fossils display extremely thin laminae, maximizing light capture in understories shaded by towering conifers. Specialized chloroplast arrangements, evidenced by exceptionally thin cuticles and high stomatal densities, suggest these plants optimized gas exchange in cool, damp air. Fossilized spores recovered from sediment cores show bursts of fern proliferation after volcanic ash deposits, hinting at rapid colonization of disturbed soils and creating lush patches that underpinned herbivore nurseries. The emergence of early flowering plants, or angiosperms, in polar regions added a new dimension to high-latitude ecology. Pollen grains of magnolid relatives and primitive eudicots appear in Alaskan and Antarctic deposits, marking the spread of these efficient photosynthesizers into polar woodlands. Their smaller, thinner leaves required less energy to grow and may have allowed for extended leaf retention during fleeting summers. Whether dinosaurs selectively browsed these novel plants for their nutrient-rich leaves remains unknown, but the co-occurrence of angiosperm pollen and juvenile dinosaur remains suggests a complex interplay between evolving plant lineages and herbivore diets. In some fossil floras, evidence emerges of understory shrubs with broad leaves and low stature, precursors to modern wintergreens. Cuticle analysis reveals thick, waxy coatings likely used to minimize water loss during polar dry spells and freeze-thaw cycles. These shrubs may have provided critical winter forage for small ornithopods and theropods, their evergreen nature offering a rare food source when deciduous fronds lay buried beneath snow. The adaptations of these plants mirror those of modern tundra communities, yet they evolved millions of years earlier under a greenhouse climate. Paleosol studies, ancient soil profiles, shine light on nutrient cycling in polar forests. Horizons rich in organic matter point to extensive leaf litter accumulation each autumn, fueling microbial communities that release nitrogen and phosphorus upon thaw. Dinosaurs foraging in the spring would have encountered nutrient-rich soils and sprouts emerging from decomposed organic layers. The timing and intensity of these soil nutrient pulses likely shaped dinosaur migration and nesting patterns, anchoring herbivore life cycles to plant-driven resource peaks. Marine and freshwater algae also contributed to polar primary productivity. Diatom and green algae fossils in sedimentary layers of coastal floodplains reveal blooms during spring meltwater surges. Such algal mats would have drawn aquatic, herbivorous dinosaurs and early turtles to feed along riverbanks, supplementing terrestrial diets. The coupling of terrestrial and aquatic plant communities created a mosaic of feeding niches, enabling polar dinosaurs to exploit multiple trophic pathways in a landscape of seasonal extremes. Interactions between plant and dinosaur communities may also have driven mutualistic relationships. Some conifer pollen grains show damage consistent with insect feeding, suggesting insect-plant dinosaur webs where beetles and other herbivores processed foliage, then small dinosaurs preyed on these insects. The resulting nutrient redistribution, through feces and carcass decay, would have fertilized soils, enhancing plant growth in a positive feedback cycle. While fossil evidence for direct mutualism is scant, such ecological intricacies hint at complex polar ecosystems beyond simple plant-herbivore dynamics. The spread of polar flora highlights the role of paleoclimate in shaping evolutionary trajectories. During greenhouse intervals, temperatures at high latitudes reached as warm as temperate zones, allowing lush forests to expand poleward. Yet even under these warmer conditions, polar light regimes imposed unique selective pressures, favoring plants capable of rapid photosynthetic responses and dormancy mechanisms. As dinosaurs adapted to these rhythms, so too did plants, their parallel evolutions weaving a shared narrative of survival beneath endless days and nights. Ancient polar forests, preserved in wood, leaf, and pollen fossils, stand as silent witnesses to a world both alien and familiar. Their adaptations to light extremes, cold spells, and nutrient cycles laid the foundation for the dinosaurs that fed, nested, and thrived among them. Yet many mysteries remain. How exactly did leaf physiology adjust to extended darkness, and which plants formed critical keystone species in polar food webs? As new plant fossils emerge from remote latitudes, the story of ancient polar flora promises to deepen, illuminating the green heart of a world poised between sunlight and shadow. Part 18. Light-driven behavior. In polar realms where sunlight waxed and waned with the seasons, dinosaur behavior became intricately tied to the rhythms of light. Trackways in Alaskan sediments sometimes reveal herds of hadrosaurs moving en masse at dawn's first glow, suggesting that migration to feeding grounds coincided precisely with the return of daylight. These synchronized movements likely maximized grazing during short summers and minimized exposure during dimmer hours. Yet the fossil record's freeze-frame moments leave open how daily activity patterns shifted throughout the year, from nearly continuous foraging in midsummer to strategic sheltering and minimal movement in the dark. Bone histology offers clues to daily and seasonal activity. In ornithopod limb bones, evidence of cyclical remodeling suggests intense wear during months of high activity, tapering off as daylight declined. Such patterns hint at an internal circadian clock, capable of adjusting metabolism and movement in response to changing light. Whether dinosaurs possessed pineal-like organs for light sensing remains unproven, but comparative anatomy with modern birds and reptiles reveals structures in the brain case that could have performed similar functions, tuning physiology to light availability and enabling survival under months of near-constant darkness. Predatory behavior too responded to light cycles. Theropods with enlarged sclerotic rings in their eyes imply adaptations for low-light hunting, yet they were forced to adjust tactics as dawn turned to dusk. During extended twilight, these predators likely shifted from vision-based ambushes to reliance on auditory and olfactory cues, as indicated by enlarged inner ear structures and extensive olfactory bulb impressions in some skulls. Such sensory flexibility would have allowed them to hunt small mammals, reptiles, or scavenged carcasses under the pale glow of the midnight sun or in the depths of polar gloom. Nesting and breeding behaviors also followed light-driven timing. Dinosaur eggshell porosity in polar deposits indicates incubation windows aligned with peak sunlight, months, ensuring embers of heat reached embryos. Adult dinosaurs may have used photoperiod cues, changes in day length, to trigger hormonal shifts, initiating nesting activities precisely when the environment could support offspring. Similar hormonal mechanics underlie modern bird reproduction, yet pinpointing these processes in dinosaurs relies on correlating nesting sites with stratigraphic layers that record seasonal light regimes. Feathered dinosaurs likely displayed plumage and courtship behaviors associated with light intensity. In dromaeosaurs, elongated forelimb feathers might have served as visual signals during the height of summer brightness, their iridescent or patterned surfaces shimmering in angled light. As days shortened, these displays likely diminished, supplanted by less conspicuous behaviors. The fossil preservation of melanosome patterns in mid-latitude specimens hints at such seasonal coloration changes, raising the possibility that polar species similarly adjusted plumage appearance for breeding and camouflage under shifting daylight. Social behaviors such as herding and territory establishment were likewise guided by light. Ethnological studies show that trackways converge at favored watering sites at predictable times, suggesting that dinosaurs used the position of the sun to navigate long distances. In the polar context, navigation by sunlight required adjustments during continuous daylight. Dinosaurs may have followed solar arcs to orient themselves, while in total darkness, they might have relied on landmarks or even geomagnetic cues. The technological sophistication needed to decipher polar dinosaur navigation remains speculative, but underscores their complex interaction with light. Physiological adaptations to light extremes extended beyond behavior into metabolism. In winter, polar dinosaurs may have entered torpor-like states, down-regulating activity and conserving energy until the return of glimmers on the horizon. Evidence of slowed bone growth and minimal remodeling during dark months supports this theory. Torpor could have ranged from daily rest cycles to multi-month dormancy, an enigmatic survival strategy that allowed these creatures to weather resource scarcity without migrating. The abrupt shifts in light during equinoxes likely triggered dramatic reprioritizations of behavior. Fossil pollen layers record explosive plant growth following the return of sunlight, and dinosaur feeding marks in vegetation fossils coincide with these pulses. This tightly coupled interplay between light-driven plant phenology and herbivore foraging strategies suggests that dinosaurs had acute sensitivity to light changes, fine-tuning reproductive, migratory and feeding behaviors to the solar calendar. The precision of these adaptations continues to amaze, providing a model for ecological synchronization in extreme environments. Ultimately, light drove the daily and seasonal lives of polar dinosaurs, weaving a tapestry of behaviors attuned to the rhythms of the sun. From foraging migrations and sensory adaptations to reproductive timing and social displays, these ancient creatures orchestrated their existence around the pulsating cycle of light and dark. Though the fossil record captures only fragments of this symphony, each bone, trackway, and nest site resonates with echoes of a world defined by the polar sun's relentless dance. Part 19. Frozen excavation. In the desolate expanses of polar plateaus, dinosaur fossils lie entombed beneath layers of permafrost, their retrieval as much a triumph of logistics as of paleontology. Teams battling sub-zero temperatures and unpredictable weather must first locate promising outcrops by interpreting satellite imagery of exposed rock in coastal cliffs. Once a site is chosen, the exposed face often hides a frozen matrix so hard that even pneumatic chisels struggle to chip away the icebound sediment. This initial phase of frozen excavation demands a blend of heavy machinery, heated shelters and painstaking manual work, each step fraught with risk and reward. Field camps operate on a razor's edge between progress and peril. Tents pitched on snowfields serve as base camps, but gale force winds and sudden whiteouts can strand workers for days. Yet in the thin polar air, daylight extends into continuous illumination in summer, providing precious hours to hammer and scoop through icy layers. As excavators reach fossiliferous beds, they employ portable steam hoses to thaw permafrost, a technique that risks bone damage if not controlled carefully. The thin line between melting enough sediment to free a bone and overheating the specimen tests both skill and nerves. Once bones emerge from the ice, they often bear a glistening sheen of mineralized fluids, preserving details invisible in drier locales. Cartilage impressions, root traces, and even skin texture can survive thanks to the permafrost's cryogenic stability. Yet removal of these delicate features requires on-site consolidation with diluted resins, a process complicated by cold that causes rapid resin thickening. Field technicians develop custom low-temperature adhesives, blending chemical ingenuity with paleontological care to stabilize specimens for transport. Transporting fossils from remote polar sites introduces its own perils. In Alaska, military helicopters lift plaster-jacketed bones from cliffside camps at dawn, navigating unpredictable gusts over jagged ridges. In Antarctica, tracked vehicles haul insulated crates across sastrugi, wind-sculpted snow ridges, while avoiding hidden crevasses. Even minor jolts can transmit fractures through ancient bones, so crates are padded with foam and suspended within larger containers. The journey from frozen field to distant laboratory is a testament to human determination and engineering under extreme conditions. Laboratory thawing protocols echo the strategies of field consolidation. Specimens slowly acclimate in temperature-controlled rooms, often over weeks, to avoid thermal shock that can crack fossil and matrix. Surrounding sediment, too, can contain delicate microfossils and pollen grains, so core samples taken during excavation are catalogued and stored for subsampling. These matrix parcels may yield insights into ancient polar flora and microfauna, preserving the broader ecological context of each dinosaur find. UV light and 3D scanning technologies play critical roles once bones thaw. Frozen excavation often reveals unexpected bone associations, vertebrae entwined with eggshell fragments, footprints preserving claw impressions in icy mud. Each detail precious for reconstructing polar dinosaur life. High-resolution scans capture surface textures and breakage patterns, creating digital backups before any further mechanical cleaning. These virtual fossils enable researchers worldwide to study specimens without risking damage to the originals. Conservation challenges extend beyond physical stability. Fungi and bacteria dormant in permafrost can activate once specimens warm, threatening to colonize and degrade organic residues. To counter this, curators treat fossils with biocidal rinses and store them in low-humidity, temperature-regulated vaults. Monitoring sensors track environmental fluctuations, ensuring that each fragment remains as pristine as when it emerged from its cryogenic sarcophagus. The very act of frozen excavation reshapes scientific questions. Rediscoveries of bone beds long thought lost under advancing ice spur new theories of glacial cycles and fossil preservation. Each freshly liberated bone forces paleontologists to reassess timelines and paleo-environmental models, as permafrost conditions can preserve specimens across extended intervals better than any temperate counterpart. The porosity of past climates and the resilience of biological archives intertwine in every icy find. Frozen excavation stands at the intersection of adventure and science, demanding perseverance in the face of sub-zero adversity. Each bone recovered from the ice is not just a fossil, it is a time capsule, a fragment of a world both alien and profound. As techniques refine and climate change exposes new outcrops, the narrative of polar dinosaurs will continue to unfold, each excavation carving deeper into the frosted annals of deep time. Part 20. CT Imaging. Advances in computed tomography have transformed our understanding of polar dinosaur anatomy, allowing researchers to peer inside fossils without damaging them. High-resolution CT scanners reveal internal structures, bone density variations, pneumatic cavities, and pathological lesions hidden beneath thick permafrost concretion. In mummified hadrosaur specimens, Connecticut imaging uncovers preserved cartilage layers and muscle attachment scars, guiding reconstructions of musculature and movement. These digital fossils open new windows into soft tissue anatomy, long thought irretrievable from polar specimens. CT scans of theropod skulls recovered from Arctic deposits illuminate cranial air sacs, extensions of the respiratory system that lightened skull weight and aided heat exchange. In high-latitude environments, efficient respiratory cooling was less critical than heat retention, suggesting that these air sacs also functioned in warming inhaled air. By mapping the three-dimensional distribution of these pneumatic chambers, paleontologists can infer breathing mechanics and thermal regulation strategies, offering a glimpse into how dinosaurs maintained stable body temperatures amid frigid air. The internal architecture of sauropod vertebrae from Antarctic sites reveals complex trabecular networks that once supported massive bodies. CT imaging quantifies bone wall thickness and internal voids, enabling biomechanical modeling of load distribution. These scans demonstrate that polar sauropods evolved reinforced vertebral structures, potentially a response to denser bone required for buoyancy control on soft, waterlogged ground. Such subtle variations in internal bone morphology highlight region-specific adaptations that would have remained concealed without non-destructive imaging. In juvenile dinosaur specimens, Connecticut imaging tracks growth patterns by visualizing successive layers of bone deposition and resorption. Growth rings appear as alternating densities within cortical bone, and CT allows measurement of their spacing and thickness without sectioning the specimen. This method has revealed accelerated growth rates in young polar dinosaurs during the sunlit summer, followed by distinct pauses correlating with winter dormancy. The ability to study ontogenetic trajectories in situ enhances our grasp of life history strategies in extreme climates. CT scans have also exposed hidden pathologies. In an Arctic Pachyrhinosaurus skull, a CT slice uncovered an internal abscess cavity behind the nasal boss, an infection that likely swelled soft tissues and altered the animal's sense of smell or pressure regulation. The unseen lesion, invisible from the exterior, underscores the power of CT to detect ancient diseases. Identifying such internal pathologies broadens our view of dinosaur health and resilience, revealing episodes of suffering and survival recorded deep within bone. Feathered dinosaur fossils benefit from CT imaging as well. By scanning carbonaceous residues around feather impressions, researchers can discern feather follicle bases and even melanosome packing arrangements. In polar sites where mineralization preserves microscopic details, Connecticut reconstruction of feather microstructure sheds light on insulation and display features. These data refine our understanding of how plumage contributed to thermal control and social signaling under shifting daylight regimes. Beyond individual specimens, Connecticut mesh-based Digital archives enable comparative studies across institutions. Digital scans of polar dinosaur fossils can be shared globally, allowing paleontologists to conduct virtual dissections and morphometric analyses in 3 dimensions. This collaborative approach accelerates research on rare, fragile specimens that cannot be transported. Through shared digital libraries, Connecticut Imaging fosters a new era of open paleoscience, connecting field sites under ice to labs around the world. Challenges remain: the high mineral density of permafrost-preserved bones sometimes attenuates X-rays, requiring adjusted scanning protocols and specialized filters. Large specimens, too, may exceed the dimensions of standard medical CT machines, necessitating portable industrial scanners or strategic sectioning prior to scanning. Overcoming these obstacles demands creative engineering solutions and interdisciplinary collaboration, blending paleontology with imaging technology expertise. As CT imaging continues to advance, offering higher resolution, faster processing, and enhanced contrast agents, the hidden interior worlds of polar dinosaurs will come into ever sharper focus. Each digital slice unearths anatomical, physiological, and pathological secrets, illuminating how these creatures thrived at Earth's extremes. The frozen fossils under CT's gaze stand poised to reveal their deepest mysteries, pixel by pixel, reshaping our view of dinosaur life in the polar outposts of deep time. Part 21. Molecular Traces. Exceptional preservation in polar permafrost offers fleeting glimpses of molecular remnants long thought to vanish with time. Collagen peptides, the fibrous proteins providing tensile strength in bone, have been detected in small but telling fragments from Arctic hadrosaur specimens. While full sequences remain elusive, mass spectrometry reveals amino acid patterns matching those of modern birds and reptiles, suggesting evolutionary continuity in structural proteins. These molecular echoes hint at the possibility of reconstructing aspects of dinosaur physiology, from growth rates to metabolic pathways, embedded in the very fibers of their fossilized bones. Even more tantalizing are reports of heme-derived porphyrins, the chemical cause of hemoglobin, preserved within the vascular canals of polar dinosaur fossils. Though degraded, these trace molecules fluoresce under ultraviolet light, indicating that blood-derived compounds can survive under near-constant cold. If corroborated, such findings could reveal how oxygen transport evolved in dinosaurs adapted to low oxygen or low temperature extremes. The endurance of these molecules, frozen in time, poses a provocative question: what other biomolecules, enzymes, hormones, or pigments might await discovery in the molecular shadows of Mesozoic remains? DNA, however, remains the holy grail that has thus far evaded retrieval from polar dinosaurs. Despite the stabilizing effects of permafrost, the half-life of DNA suggests that no intact strands could endure beyond a few million years. Yet fragments of degraded genetic material may linger in microscopic pockets, detectable only by ultra-sensitive techniques. Novel approaches, such as targeted enrichment or nanopore sequencing of ultra-short fragments, offer faint hope that trace genetic information could be uncovered, rewriting the limits of paleogenomics and deepening our grasp of dinosaur evolution. Lipids, the fats and oils integral to cell membranes, also show promise for deep time preservation. In permafrost-preserved bones, thin layers of organic residue sometimes yield lipid biomarkers, molecular fossils that resist degradation. These residues can differentiate tissue types, distinguishing cartilage from bone or muscle, and hint at the presence of ancient cell membranes. Lipid analyses could therefore map soft tissue distribution in fossils, revealing the anatomical layout of organs long since lost to decay, a molecular blueprint concealed within mineralized bone. Pigment molecules, such as melanin-derived eumelanin and pheomelanin, survive in fossil feathers through stable melanosome structures, but chemical traces of these pigments occasionally remain. High-latitude dinosaur feathers examined under Raman spectroscopy show weak but distinct signals corresponding to aromatic rings found in melanin. These molecular fingerprints corroborate melanosome-based color reconstructions and suggest that polar dinosaurs bore vivid plumage colors, perhaps tuned to absorb heat or communicate under polar light extremes. Enzymatic cofactors and small metabolites, though highly susceptible to breakdown, might leave behind modified signatures in fossil bone. Advanced techniques like time-of-flight secondary ion mass spectrometry, TOF-SIMS, can detect trace organic fragments, mapping their spatial distribution within bone microstructure. Although definitive identification of specific metabolites remains on the horizon, the detection of patterns consistent with ancient metabolic byproducts would revolutionize our understanding of dinosaur biochemistry, offering a direct link to cellular processes. Molecular taphonomy, the study of how biomolecules transform after death, reveals that permafrost's cold anoxic conditions slow hydrolysis and microbial degradation, extending the window for molecular survival. Yet freeze-thaw cycles and mineralization can still alter chemical signatures. Understanding these diagenetic pathways is critical for distinguishing authentic ancient molecules from contaminants. Researchers use experimental diagenesis, simulating long-term burial in laboratory settings, to validate molecular findings, ensuring that claimed dinosaur biomolecules truly reflect deep-time preservation. Collaborations between paleontologists and biochemists have created interdisciplinary frameworks to hunt these molecular vestiges. Field protocols now include swabbing bone surfaces for soluble organics before consolidation, preserving potential biomolecules that traditional excavations might wash away. Laboratory workflows emphasize contamination control and employ multiple analytical platforms, GCMS, LCMS, Raman spectroscopy, to triangulate molecular identities. This methodical, multi-pronged approach is gradually unveiling the faint molecular whispers embedded in polar dinosaur remains. Though still in its infancy, the search for molecular traces in polar dinosaurs opens doors to new realms of paleobiology. Each detected peptide, lipid biomarker, or pigment fragment peels back another layer of time, hinting at living processes preserved in stone and ice. As techniques advance and fresh specimens emerge from thawing permafrost, the molecular secrets of polar dinosaurs promise to emerge from dormancy, illuminating their physiology, behavior, and evolutionary heritage in unprecedented detail. Part 22. Behavior models. Computer simulations and biomechanical reconstructions are illuminating how polar dinosaurs might have behaved within their frozen realms. By integrating trackway patterns, skeletal dimensions, and environmental parameters, researchers build virtual herds that move, forage, and rest according to programmed rules. In one model of hadrosaur flock movement, agents respond to diminishing daylight by clustering closer together, reducing individual energy expenditure. These digital behaviors echo fossil evidence of tight trackway groupings in autumn layers, suggesting that real herds may have adopted compact formations to conserve warmth during seasonal transitions. Agent-based models of predation in polar woodlands reveal how apex predators like Nanosaurus could have optimized ambush strategies under low-light conditions. Virtual predators navigate toward areas of high prey density, riparian zones, and hatchling nurseries while using terrain features for concealment. When simulated under Arctic light regimes, Nanosaurus agents exhibit increased nocturnal activity and ambush frequency during dawn and dusk periods, aligning with hypotheses drawn from sclerotic ring analysis. These behavior models offer plausible scenarios for predator-prey interactions that left lasting traces in the fossil record. Reproductive behaviors also come into focus through modeling. By correlating nesting ground locations with paleobotanical reconstructions, virtual dinosaur agents can select optimal nesting sites based on factors like solar incidence, substrate temperature, and predator access. In simulated Arctic summers, nesting agents cluster in geothermally warmed soils, analogous to modern megapode strategies, highlighting the potential use of microhabitats to incubate eggs. Such models help explain why polar nesting sites often coincide with ancient volcanic vents or warm spring outflows. Physiological models layered into these simulations reveal how metabolism and thermoregulation influence daily rhythms. Dinosaurs with high metabolic rates require frequent feeding. Virtual ornithomimids in polar models increase foraging range during continuous daylight, yet curtail activity below threshold temperatures. Incorporating bone histology, inferred metabolic parameters, allows these agents to enter torpor states during winter, reducing simulated energy demands. The emergence of such resting states supports the idea that polar dinosaurs combined active and quiescent periods to navigate extreme seasonal cycles. Social interaction models explore how group size and structure adapted to resource scarcity. Simulations of mixed-age herds indicate that including juveniles increases collective vigilance but also heightens food competition. When modeled under winter resource constraints, herds split into age-segregated subgroups, juvenile nurseries foraging on low-lying vegetation, while adults range more widely. These patterns echo fossil bone beds showing age-segregated assemblages, suggesting that modeling can replicate real-life coping strategies in harsh polar ecosystems. Migration models, though challenging at high latitudes, demonstrate how partial migrations might occur. By linking virtual agents to geospatial maps of seasonal vegetation distribution, some herbivore agents relocate hundreds of kilometers to follow thaw lines, while others remain stationary. These hybrid strategies, residency and short-range movement, maximize survival odds without requiring the epic migrations once hypothesized. Such nuanced movement patterns align better with the fragmentary trackway and bone bed data than blanket all-or-nothing migration theories. Predicted sensory behaviors emerge from models incorporating visual and olfactory parameters. Simulated Trudon agents adjust hunting tactics according to light availability, shifting from vision-based stalking in summer to scent tracking in winter. Incorporating data on olfactory bulb size and nasal cavity volume, these models suggest that sensory trade-offs shaped seasonal foraging techniques, a hypothesis that meshes with tooth-wear patterns, indicating omnivory and opportunistic feeding under diverse conditions. Comfort and sheltering behaviors also appear in virtual reconstructions. Some polar dinosaur agents seek microtopographic features, sun-warmed slopes or downwind lee sides, for resting during extreme cold. Recreating wind patterns and insulation in digital landscapes reveals how even minor terrain variations could create thermal refuges. These microhabitats, invisible in regional maps but apparent in detailed topographic data, may explain isolated fossil finds in unexpected sedimentary contexts. Through sophisticated behavior models, the frozen history of polar dinosaurs comes alive in pixels and algorithms. These virtual experiments, grounded in fossil evidence and environmental data, generate testable predictions about movement, feeding, reproduction, and sociality under extreme light cycles. As models refine and incorporate new discoveries, they promise to transform the static record of bones and tracks into dynamic chronicles of dinosaur life at Earth's icy extremes. Part 23. Museum displays. Exhibiting polar dinosaurs requires more than glass cases and placards. It demands immersive experiences that convey the drama of ancient extremes. Museums housing Arctic and Antarctic specimens craft dioramas that recreate snow-dusted conifer forests under perpetual daylight or the haunting blue twilight of polar dusk. Full articulated skeletons of Cryolophosaurus are posed mid-stride on frosted bases, their ribs and vertebrae reconstructed to hint at the fierce winds they once faced. Carefully engineered LED lighting cycles mimic seasonal daylight changes, allowing visitors to sense how light-shaped dinosaur rhythms. Real permafrost excavated specimens often arrive encrusted in frozen matrix, a visual testament to their millennia-long entombment. Rather than fully cleaning these fossils, some institutions choose to display them in partial jackets, sections of rock and ice left intact, to illustrate field conditions and preservation contexts. Accompanying multimedia exhibits show video of steam hoses and heated tents at excavation sites, bridging the gap between lab and field. These as-found specimens captivate audiences by connecting them directly to the cold climate challenges of paleontology. Interactive digital displays play a pivotal role in portraying life beneath polar light regimes. Touchscreens allow visitors to manipulate CT scan datasets, slicing through hadrosaur femora or troodontid skulls to unveil hidden pneumatic chambers. Augmented reality apps overlay mummified skin impressions onto skeletons, revealing subtle textures and coloration. By exploring these virtual layers, museum-goers glimpse the internal anatomies and soft tissue mysteries that permafrost preservation has uniquely safeguarded. Soundscapes further immerse visitors in polar worlds. Recordings of howling katabatic winds and subtle bird calls, recreated by blending modern Arctic avian vocalizations, echo through galleries, evoking the environmental backdrop that shaped dinosaur physiology and behavior. Subtle vibrations beneath flooring simulate rumbling conga of migrating herds or the thump of massive tails against ice-embedded ground. These multisensory elements underscore the dynamic interplay between creatures and their frostbound habitats. Educational programming extends beyond static displays. Workshops enable participants to handle real permafrost blocks, chipping away matrix with miniature steam jets while guided by paleontologists. Children's programs include molding dinosaur icicles, making plaster impressions of trackway molds, and mapping seasonal migration routes on polar maps. These hands-on experiences illuminate the scientific techniques behind frozen excavation and inspire a deeper appreciation for the perseverance required to uncover polar fossils. Conservation labs visible through museum glass enclose ongoing preparation of permafrost specimens. Visitors watch as technicians apply low-temperature consolidants, digitally document microscopic bone histology, and scrub specimens under lamps set to just above freezing. Transparent cases demystify the post-excavation process, illustrating the fragility and care needed to preserve molecular traces. This behind-the-scenes access fosters respect for the painstaking work that brings frozen fossils to public view. Traveling exhibitions showcase polar dinosaur casts and interactive modules, bringing frozen worlds to regions far from the poles. These mobile displays feature full-scale trackway replicas embedded in faux permafrost, allowing schools and small museums to host immersive polar paleontology experiences. Accompanying curricula emphasize climate change parallels, how modern permafrost thaw endangers both living ecosystems and fossil archives, tying prehistoric lessons to contemporary environmental concerns. Collaborations with Inuit and Sami communities integrate indigenous knowledge into exhibits, highlighting human survival under polar light cycles and drawing parallels with dinosaur adaptations. Storytelling sessions by indigenous elders detail animal migrations and seasonal resource use, creating cross-cultural dialogues about resilience. These partnerships enrich museum narratives, reminding audiences that adaptation to light extremes is a shared thread across deep time. Museums play a vital role in translating the mysteries of polar dinosaurs into accessible stories of discovery and survival. Through innovative displays, interactive technologies, and community engagement, they bring to life the frozen saga of dinosaurs at Earth's extremes. Each exhibit, whether framed by ice-mimicking glass or pulsing with digital reconstructions, invites audiences to consider how light, cold, and time conspired to forge remarkable adaptations, and how these stories still resonate in a warming world. Part 24. New Technologies. Emerging technologies are revolutionizing paleontological research in polar regions, transforming how fossils are discovered, excavated, and analyzed. Unmanned aerial vehicles equipped with LiDAR sensors now map ice and snow-covered terrains, identifying subtle geomorphic breaks that hint at buried fossil-bearing layers. High-resolution elevation models generated by these drones guide field teams to prime excavation sites, reducing the time spent slogging through treacherous ice fields. The precision of LiDAR mapping has already led to the discovery of previously unknown bone bed exposures hidden beneath thin snowdrifts. Thermal imaging aboard aerial platforms reveals subsurface heat anomalies, areas where permafrost is disrupted by decomposing organic matter or subsurface cavities. Such hotspots often coincide with fossil-rich pockets that absorb and retain heat differently than surrounding sediment. By flying transects at dawn, when temperature contrasts are greatest, researchers pinpoint excavation targets more efficiently than ever. This method reduces environmental impact by focusing efforts on promising locales and minimizing unnecessary disturbance of fragile polar landscapes. In the laboratory, synchrotron radiation facilities enable non-destructive chemical mapping of permafrost fossils at micron scales. Researchers beam intense X-rays through fossilized bones to trace the distribution of elements like phosphorus, calcium, and trace metals. These elemental maps reveal directional growth patterns, diagenetic changes, and even hotspots of preserved organic compounds. The insights gleaned guide sampling strategies for molecular analyses, ensuring that scarce biomolecular residues are extracted from the most pristine regions of bone. Portable mass spectrometers, once confined to specialized labs, now travel with field teams. These instruments can analyze trace organic compounds, lipids, pigments, and potential protein fragments on-site, providing immediate feedback on fossil preservation quality. Rapid screening informs decisions about which specimens to prioritize for detailed excavation and laboratory study. The portability of these devices shortens the feedback loop, allowing chemists and paleontologists to collaborate in real time under polar skies. Robotic excavators designed for freezing temperatures are under development, promising to automate the initial removal of overlying permafrost. Equipped with thermal drilling heads and precision articulation, these robots could tackle the most labor-intensive excavation phases, reducing human exposure to extreme cold and minimizing risk. Early prototypes demonstrate the feasibility of robotic trenching in permafrost, hinting at a future where machines prepare fossil-bearing layers under human supervision, improving both speed and safety. Machine learning algorithms sift through massive datasets, satellite imagery, trackway photos, sediment analyses, to predict new fossil locales. By training on known fossil site attributes, these AI models identify geological signatures correlated with successful discoveries. This predictive modeling prioritizes survey areas, optimizing logistics for costly polar expeditions. Early trials in Arctic and Antarctic archives show a significant uplift in site detection rates, suggesting that AI will soon become an indispensable tool in the polar paleontologist's kit. In situ 3D printing of protective field jackets is becoming a reality. Lightweight composite materials can now be extruded directly onto exposed fossils, conforming precisely to bone surfaces and surrounding matrix. These custom jackets eliminate bulky plaster and burlap wrappings, reducing shipment weight and improving specimen stability. Field teams report that 3D printed jackets cure faster and require fewer materials, streamlining the preservation process in environments where supply lines are tenuous. Virtual reality reconstructions created from integrated CT, LiDAR, and photogrammetry data enable researchers and the public alike to explore polar dig sites remotely. By donning a VR headset, one can walk among frozen bone beds, inspect delicate skin impressions, and observe the interplay of geology and paleontology in immersive detail. These digital twin environments facilitate collaboration among international teams year-round, transcending the brevity of polar field seasons and democratizing access to rare fossil contexts. As research pushes further into the extremes of Earth, new technologies continue to emerge, each one a key to unlocking the secrets buried beneath ice and stone. From drone-assisted discovery to molecular screening in the field, these innovations accelerate paleontological breakthroughs and safeguard fragile specimens. In the crucible of polar exploration, technology and tenacity unite, ensuring that the frozen stories of dinosaurs endure for generations to come. Part 25. Next expeditions. Polar paleontology stands on the cusp of new frontiers as melting permafrost and receding ice unveil fossil-rich deposits long hidden beneath frozen layers. Future expeditions target remote sectors of the transantarctic mountains, where recently exposed late Jurassic outcrops may harbor ancestral dinosaur lineages. Equipped with augmented reality mapping and drone reconnaissance, teams will chart previously inaccessible cliff faces, following hints of bone fragments in glacial meltwater channels. The promise of uncovering new taxa, perhaps the earliest known polar sauropods or feathered theropods, drives a renewed spirit of exploration into these extreme environments. In Arctic Canada, thawing coastal bluffs along Ellesmere Island beckon scientists to investigate late Cretaceous strata that have yielded only fragmentary remains to date. Coordinating with indigenous communities, interdisciplinary teams will blend traditional environmental knowledge with geophysical surveys to locate fossil hotspots. Portable labs established on sea ice will enable rapid processing of specimens, minimizing degradation risks during transport. These collaborative approaches aim to fill gaps in how northernmost dinosaurs responded to high-latitude climate changes and ecological pressures at the end of the Mesozoic. Beyond mere discovery, future expeditions will emphasize conservation and digital archiving. Satellite-enabled field notebooks, 3D photogrammetry, and blockchain-based specimen tracking will ensure that each fossil's context is preserved in perpetuity. As specimens move from site to museum, a permanent digital twin remains accessible to researchers worldwide. This fusion of exploration and preservation establishes a blueprint for scientific stewardship, ensuring that the history hidden in permafrost survives environmental and societal changes. Technological integration will extend to citizen science initiatives. Backpack-mounted GPS scanners and open-source field apps will empower amateur fossil hunters accompanying professional teams. By crowdsourcing documentation of bone fragments and vegetative anomalies, these programs accelerate site surveys and foster public engagement. Remote live feeds from polar camps, coupled with interactive Conda sessions, will bring the thrill of discovery to classrooms and living rooms around the globe, building a new generation of polar paleontologists. Experimental in situ analysis will push the limits of field-based science. Mobile mass spectrometers and portable DNA sequencers will test for molecular signatures of proteins and genetic fragments on site, enabling immediate decisions about specimen value and storage. These innovations promise to reduce sample loss and contamination, slashing the time between discovery and data acquisition. By bringing the molecular lab to the rock face, future expeditions will capture fleeting biomolecular clues before they vanish with thaw and decay. Climate modeling teams will join paleontologists on next expeditions, drilling sediment cores adjacent to bone beds to reconstruct past temperature, precipitation, and atmospheric composition. Layered together, climatic and biological records will illuminate how polar ecosystems shifted through greenhouse peaks and cooling trends. These integrated studies not only enrich our understanding of dinosaur habitats, but also offer analogues for modern climate dynamics, linking deep-time lessons to present-day concerns about polar thaw and biodiversity loss. Logistical innovations promise to expand the reach of polar research. Solar-powered snow vehicles capable of traversing sastrugi and thin ice will transport personnel and cargo quietly and with minimal carbon footprint. Autonomous submersibles deployed under sea ice will scout underwater bone beds, potential repositories of marine reptiles and submerged dinosaur trackways. These vehicles will map seafloor geology in 3D, uncovering new fossil sites along paleo shorelines now hidden beneath frigid waters. International cooperation will define the next era of polar discoveries. Shared research stations co-funded by multiple nations will offer year-round access to key sites, reducing the window of opportunity imposed by harsh seasons. Collaborative networks will standardize excavation, conservation, and data protocols, fostering seamless integration of findings across borders. This global partnership ensures that the study of polar dinosaurs transcends political boundaries, united by the common pursuit of knowledge about life in Earth's coldest realms. Ultimately, the next expeditions promise not only to fill gaps in the dinosaur fossil record, but also to inspire wonder at the adaptability of life. From the first bone fragments revealed by melting ice to complete articulated skeletons sheltered by new shelters, each discovery will reshape our view of polar dinosaurs as denizens of a dynamic, ever-changing world. As scientists venture further into the frost, the frozen narratives of these ancient giants will continue to unfold, reminding us that even at the poles, life endures in splendid variety. Part 26. Ice Sheet Dynamics. The ever-shifting rhythms of polar ice sheets sculpted the landscapes that dinosaurs once traversed, their advance and retreat patterns driving ecological turnover and influencing fossil preservation. In the late Cretaceous, ice was largely absent, but nascent glacial processes began sculpting high-latitude topography. River valleys, originally carved by warm season meltwater, later experienced episodic freeze events, creating permafrost that would, tens of millions of years later, encapsulate dinosaur remains. Understanding these early ice dynamics helps explain why some bone beds lie trapped beneath ancient frost lines, frozen in time by the slow creep of glacial tongues. Sedimentary records from Antarctic coastal cliffs reveal cyclical deposits of glacial till interleaved with dinosaur-bearing mudstones. These tills, mixtures of clay, sand and boulders dropped by retreating ice, often serve as markers bracketing fossil-rich layers. In some sections, a thick till layer caps a prolific bone bed, indicating that an ice advance buried the remains, preserving them under a cold, stable layer. Later ice retreats expose these deposits, offering rare windows into Mesozoic life. The interplay of advance and retreat thus not only shaped ancient habitats, but also controlled where and when fossils would resurface for discovery. Isotopic studies of oxygen and carbon in glacial varves, annual layers of sediment deposited in glacial meltwater lakes, provide climate context for dinosaur ecosystems. By measuring isotopic shifts between varves, scientists reconstruct temperature oscillations corresponding to ice sheet fluctuations. When correlated with nearby bone beds, these climate records reveal how dinosaur communities responded to environmental stress, with extinction pulses and faunal turnovers aligning with periods of glacial advance. This temporal coupling underscores the power of ice as both an ecological driver and a curator of the fossil record. Modern analogues in the Arctic show that present-day ice sheets advance and retreat on decadal to centennial scales, influenced by ocean currents and atmospheric patterns. By comparing these dynamics with ancient sedimentation rates, researchers estimate how quickly late Cretaceous permafrost could form over vertebrate remains. Rapid permafrost establishment, a matter of years rather than centuries, would have been necessary to halt decay and protect biomolecules. These insights refine excavation strategies, guiding paleontologists to search beneath specific till layers most likely to yield exceptionally preserved specimens. The geometry of paleo ice sheets also influenced dinosaur migration and distribution. As ice lobes extended into lowland corridors, they blocked traditional movement routes, forcing herds to find alternative paths or face dwindling resources. Fossil trackways sometimes end abruptly at paleoglacial margins, hinting at impassable barriers that shaped herd structure and genetic isolation. Mapping these ancient ice boundaries helps explain patterns of endemism and speciation among polar dinosaurs, revealing how climate sculpted evolutionary pathways. Underneath ice sheets, subglacial meltwater channels eroded bedrock and deposited fine-grained sediments in deep troughs. In some polar outcrops, these subglacial deposits preserve microfossils, pollen, spores, and even microbial mats that predate glaciation by millions of years. When overlaid by later bone beds, they provide a layered archive of ecosystem change from greenhouse warmth to icehouse conditions. This stratigraphic palimpsest offers a unique opportunity to study the transition from dinosaur-dominated ecosystems to those governed by cold climate organisms. Glacial static rebound, the uplift of crust after ice mass loss, also plays a role in fossil exposure. As ancient ice sheets melted, The land once depressed by glacial weight slowly rose, tilting and folding sedimentary sequences. Today, this uplift reveals cliff faces rich in dinosaur fossils previously buried beneath deeper sediments. Understanding rebound rates allows researchers to predict where emerging outcrops will appear as uplift continues, offering a roadmap for future fossil discoveries guided by geophysical modeling. The chemistry of subglacial environments impacts bone diagenesis. Meltwater rich in dissolved ions can infiltrate permafrost, precipitating minerals that either preserve or destroy organic structures. In some Antarctic locales, high concentrations of iron and silica have mineralized bone collagen, creating natural casts that retain ultrastructural details. Elsewhere, acidic meltwaters have leached bone apatite, complicating radiometric dating and isotopic analyses. Recognizing these chemical processes is essential for interpreting fossil integrity and for choosing appropriate conservation treatments post-excavation. Ultimately, ice sheet dynamics weave a complex narrative that links ancient climates, dinosaur life, and modern discoveries. The advance and retreat of glaciers shaped habitats, directed migrations, preserved fragile tissues, and revealed fossils through dynamic landform evolution. As models of paleoglaciation grow more sophisticated and remote sensing unmasks hidden till deposits, the story of icy sculptors and their prehistoric inhabitants will continue to unfold, an intertwined saga of ice and life at Earth's frozen frontiers. Part 27. Nest Predators. Ancient nesting grounds in polar regions offered sanctuary for dinosaur eggs and hatchlings, but they also attracted a host of nest predators ready to exploit this seasonal bounty. Among the most insidious were small theropods like Troodon, whose sharp, serrated teeth and nimble builds suited them to raiding shallow nests. Trackway evidence near nesting sites shows parallel looping paths, aeolian sands preserving the stealthy patrols of these opportunistic hunters. By circling nests under the cover of low-angle light, Troodon could snatch eggs and young without attracting larger carnivores, a behavior reflected in partially devoured eggshells and tooth-marked clutch barriers. Mammal-like reptiles and early mammals also prowled polar nesting sites. Fossils of small insectivorous creatures in Arctic deposits suggest that multituberculates and symmetrodonts scavenged husks and shell fragments left by larger predators. Their cheek teeth, adapted for crushing, likely allowed them to consume residual egg membranes and small hatchling remains. Though diminutive, these mammals may have formed a nocturnal scavenger guild, slipping into nests after dark to clean up leftovers, thus playing a critical role in nutrient recycling within nesting colonies. Even other dinosaurs sometimes turned on nests. In some polar bone beds, juvenile ceratopsian bones intermingle with shattered sauropod eggs, hinting at predatory forays by horned dinosaurs on smaller conspecific nesting grounds. Powerful beaks that once sheared tough vegetation became weapons for crushing eggs. Such intraclade predation underscores the ruthless pragmatism of survival. When plant matter waned in winter, horned herbivores may have resorted to opportunistic carnivory, breaching nests to access high-protein resources. Oviraptorosaurs, though more common in mid-latitudes, likely extended their range into polar ecosystems during warmer intervals. Fossil fragments bearing distinctive oviraptorid eggshells appear in some Arctic strata, their identification suggesting that these bird-like theropods nested alongside hadrosaurs and ceratopsians. Their specialized crushing beaks and robust jaws empowered them to open large eggs, and their brooding-like posture, borne out by mid-latitude finds, could conceal their own predatory intentions, luring in smaller dinosaurs before striking. Predatory insects also left their marks. Fossilized eggshell fragments exhibit tiny puncture wounds consistent with coleopteran mandibles. Ancient beetles, drawn to the protein-rich environment of nests, bored into shells to the benefit of their larvae. These micro-predators, though invisible to most eyes, shaped nesting behaviors. Adults may have covered nests with plant debris, not only for insulation, but also to mask olfactory and visual signals from insect scouts. The arms race between nesting dinosaurs and invertebrate raiders played out on microscopic battlefields, sealed in fossil record. Marine reptiles occasionally exploited coastal nesting sites. In regions where nests lay close to ancient shorelines, plesiosaurs and mosasaurs may have scavenged eggs washed into tidal flats. Tooth mark patterns on eggshell fragments mirror those found on fish bones, indicating similar feeding techniques. Lateral jaw shakes to fragment brittle shells. Such dual threats from above and below likely influenced how and where dinosaurs chose to nest, favoring inland elevations over flood-prone flats when possible. Nest predators left chemical traces as well. High concentrations of coprolites, fossilized droppings, around some nesting colonies contain crushed eggshell fragments and bone shards. Analyzing these coprolites reveals digestive acids and microbial residues indicative of carnivorous digestion. By mapping coprolite distribution, paleontologists reconstruct feeding hotspots, pinpointing which predators dominated particular nesting grounds. These nutrient-rich scatter zones in turn attracted scavengers, creating concentric rings of predation and cleanup around nests. Defensive adaptations in response to nest predation emerge in the fossil record. Some dinosaur eggshells bear thicker regions at poles or seams, perhaps reinforcing vulnerable areas against crushing. Others show evidence of covering behavior, with nest sediments preserving plant root traces woven into the clutch substrate, a primitive form of camouflage. The evolution of such defenses suggests that nest predation exerted strong selective pressures on both adult and embryonic dinosaurs, driving innovations in eggshell architecture and nesting strategies. In the frozen chapters of polar paleoecology, nest predators played pivotal roles in shaping dinosaur reproductive strategies. From stealthy theropods to opportunistic mammals, from chewing insects to marauding marine reptiles, a diverse cast of characters converged on nesting grounds, each vying for a share of the next generation. Their marks, tracks, tooth scars, chemical residues transform static bone beds into dynamic theaters of predation and defense, where survival hinged on the interplay of offense and protection under the polar sun and shadows. Part 28. Climate shifts. Polar dinosaur ecosystems were profoundly influenced by dramatic climate oscillations that transformed habitats across millennia. During greenhouse peaks in the early Cretaceous, polar latitudes enjoyed temperate climates with frost-free forests extending to the poles. Fossil flora, lush conifer canopies and diverse ferns thrived under these mild conditions, supporting robust dinosaur populations. Yet rhythms of volcanic activity and shifting ocean currents triggered gradual cooling phases, punctuated by abrupt events that plunged temperatures and altered precipitation patterns. These climatic swings dictated resource availability, shaping evolutionary pressures on high-latitude dinosaurs. Evidence from marine sediments off the Antarctic coast reveals periodic pulses of glacial activity beginning in the late Cretaceous. Isotopic analyses of foraminifera shells document oscillations between warmer and cooler seawater temperatures, correlating with shifts in floral assemblages on land. As cooler intervals became more frequent, polar forests contracted, replaced by open woodlands and scrub. Herbivorous dinosaurs adapted by diversifying diets, shifting from broadleaf browsing to consuming tougher, woody understory plants. These botanical transitions left their mark in tooth-wear patterns and jaw mechanics, highlighting one facet of climate-driven adaptation. Rapid climate perturbations, such as large igneous province eruptions, injected aerosols into the atmosphere, inducing short-term volcanic winters. Layers of volcanic ash found interbedded with polar dinosaur bone beds mark these events. Such episodes likely suppressed photosynthesis, leading to mass die-offs of vegetation and consequently herbivore populations. Carnivores reliant on these herbivores faced cascading food shortages. The survival and rebound of dinosaur communities after these crises illustrate both vulnerability to and resilience against sudden environmental upheavals. Tundra-like conditions emerged intermittently as cooling intensified, with frozen soils encroaching on ancient floodplains. Permafrost indicators, ice wedge casts and patterned ground structures appear in some late Cretaceous sedimentary layers, documenting the first onset of ground freezing. Dinosaurs confronted this emerging permafrost by altering nesting locations and foraging behaviors. Trackways crossing patterned ground suggest that some species tested frozen substrates before committing to migration or nesting, revealing a behavioral flexibility driven by climate stress. Paleobotanical records show that during cooling phases, flowering plant diversity in polar regions dwindled, replaced by gymnosperm, refugia species adapted to cold. This reduction in angiosperm abundance narrowed dietary options for herbivorous dinosaurs, forcing them to exploit alternative food sources such as seeds, bark, and evergreen needles. Such dietary shifts appear in coprolite analyses, where microscopic fragments of woody tissues and resin droplets attest to creative foraging strategies under nutritional duress. Faunal turnover accompanied climatic transitions. At several Arctic sites, the replacement of one hadrosaur species by another coincides with cooling pulses. This pattern suggests that certain species possessed thermal tolerances or dietary specializations better suited to cooler environments. Comparative studies of bone growth rates reveal that successor species matured more slowly, perhaps an adaptation to shorter growing seasons. These generational shifts underscore how climate waves sculpted dinosaur biodiversity in polar habitats. Climatic events also influenced dinosaur physiology. Bone isotopes document periods of pronounced metabolic slowdown during colder intervals, with lines of arrested growth thickening to denote prolonged stress. Conversely, rapid summer growth rings narrow as growing seasons shortened. These histological records provide a biological diary of climate impacts, capturing the toll of cooler, darker years on the internal rhythms of polar dinosaurs. As climates cooled overall toward the end of the Cretaceous, polar ecosystems entered a novel regime of pronounced seasonality, setting the stage for mammalian expansions after the dinosaurs' extinction. The gradual shift from greenhouse warmth to icehouse glaciation rewrote ecological relationships. For dinosaurs, each climate shift demanded new adaptations, behavioral, physiological, and anatomical, to survive in what became increasingly marginal habitats at Earth's frozen edges. The story of climate shifts in polar dinosaur realms offers a powerful lesson in resilience and vulnerability. Dinosaurs endured cycles of warming and cooling, volcanic winters and emerging permafrost, adapting their diets, behaviors, and life histories in response. Yet these same pressures also drove evolutionary change and ultimately extinction in the face of sustained stress. As modern climates change with unprecedented speed, the ancient record of polar dinosaurs stands as both a testament to adaptability and a warning of limits faced by even the most formidable creatures. Part 29. Saltwater crossings. Even dinosaurs, masters of land, sometimes ventured into briny realms. In the Cretaceous seas surrounding Antarctica, plesiosaurs like Marenosaurus and Hydratherosaurus patrolled nutrient-rich waters, their long necks sweeping for fish beneath ice-laced surfaces. These marine reptiles laid eggs on coastal beaches and likely hauled themselves ashore in sheltered coves. Occasional skeletal remains found inland hint at their ability to navigate estuaries and even shallow lagoons, carving paths between islands and mainland via tidal flats that connected polar archipelagos. Hadrosaurs, though primarily terrestrial, show evidence of saltwater tolerance that may have facilitated island hopping. Bone histology of Antarctic hadrosaur specimens reveals elevated chloride ion concentrations within vascular canals, an indicator that these dinosaurs drank or swam in brackish waters. Coastal trackways preserved on Seymour and James Ross Islands sometimes disappear into tidal sediments, suggesting that herds entered shallow bays, perhaps to reach offshore feeding grounds of nutrient-rich vegetation growing on small islands. These amphibious journeys would have demanded navigational acuity and physiological adjustments to saltwater ingestion. Titanosaurs, such as those related to South America's saltosaurids, may have crossed narrow seaways during periods of low sea level. Sedimentary data place land bridges between Patagonia and Antarctic Peninsula at intervals during the late Cretaceous. These transient corridors allowed massive sauropods to disperse southward, following vegetation belts. The sheer scale of these creatures crossing shallow channels, wading through tidewaters, paints a vivid image of long-necked giants forging pathways across prehistoric straits. Smaller theropods like Troodon and Dromaeosaurus show isotopic evidence of marine resource use, with trace marine carbon signatures in tooth enamel that align with known coastal deposits. This suggests they scavenged fish carcasses washed ashore or hunted near shorelines, ingesting saltwater prey and possibly venturing into shallow tidal zones. Their lightweight frames and agile bills would have enabled them to pick their way over slippery rocks and through tidal pools in search of easy meals. Marine reptiles themselves sometimes breached into freshwater systems, venturing upriver in pursuit of spawning fish. Fossilized juvenile plesiosaur remains found in deltaic deposits indicate nurseries established far from the open ocean. These freshwater forays parallel modern sea turtles' habit of nesting on distant beaches. Polar plesiosaurs may have followed seasonal fish runs into Arctic rivers or glacial meltwater streams, exploiting a diversity of prey and expanding their ecological reach. Island hopping behavior left traces in both marine and terrestrial strata. In Antarctica's La Meseta Formation, mixed assemblages of dinosaur and marine reptile remains suggest overlapping habitats on floodplains fringed by tide-influenced wetlands. Occasional sandstone lenses preserving both hadrosaur footprints and plesiosaur vertebrae hint at catastrophic events, storm surges or tsunamis that deposited terrestrial and marine organisms together. These mixed deposits testify to the proximity of land and sea in polar ecosystems, where boundaries blurred under dynamic climatic conditions. Crossing saltwater may have offered refuge from inland predators and competitors. Hadrosaurs and ceratopsians drifting on mats of vegetation, akin to modern iguanas in the Galapagos, could colonize new islands isolated from larger carnivores. Over evolutionary time, such accidental voyages may have sparked speciation events, producing endemic dwarf forms or novel adaptations to island ecosystems. While direct genetic evidence remains out of reach, the distribution patterns of related genera across southern hemisphere landmasses hint at rafting or wading as viable dispersal mechanisms. Physiological challenges of saltwater crossings were non-trivial. Dinosaurs lack modern salt-excreting glands, so desalination of ingested seawater likely relied on behavioral strategies, minimizing swimming time, drinking fresh water from puddles on driftwood, and scavenging moist vegetation. Stress from osmoregulation may have limited the duration and distance of saltwater ventures, favoring short crossings during calm weather. These constraints underscore the remarkable boldness of any dinosaur brave enough to test the boundaries between land and sea. Saltwater crossings reveal a surprising plasticity in dinosaur ecology, demonstrating that even ancient giants could exploit coastal and nearshore habitats to expand their ranges. From plesiosaurs weaving between islands to dinosaurs wading across tidal flats, these migrations illuminate the entangled histories of land and sea in polar realms. As future discoveries unearth more evidence of marine-terrestrial interplay, the narrative of polar dinosaurs will continue to deepen, charting journeys forged across the ancient waterways of a frozen world. Part 30. Legacy in Rocks. The story of polar dinosaurs does not fade with extinction. It echoes through the rocks that cradle their bones. In Antarctica and high-latitude regions of Australia and Alaska, the fossil record offers not only snapshots of ancient species, but entire ecosystems immortalized in stone. Layer upon layer of silt, ash, and sediment tells the tale of creatures that once roamed under the southern lights, enduring cold seasons, volcanic eruptions, and shifts in Earth's climate. Every fossil embedded in these layers is more than just anatomy. It is the memory of an ancient life, preserved with geologic patience. The James Ross Basin on the Antarctic Peninsula is one of the richest sources of polar dinosaur fossils. Within its strata lie bones from hadrosaurs, ankylosaurs, and theropods, all entombed in Cretaceous sediment laid down when the continent was lusher and milder than today. These fossils are found alongside impressions of leaves, pollen grains, and freshwater invertebrates, revealing a once verdant floodplain teeming with life. Volcanic ash layers within the same formation serve as precise timestamps, enabling researchers to map evolutionary events and extinction pulses with astonishing clarity. Fossil footprints etched into the ancient stone of Victoria, Australia, tell of passage through forests drenched in half a year of twilight. These trace fossils often preserve the direction, speed, and even behavior of dinosaurs, such as herds turning sharply to avoid an unseen predator, or pausing near what was once a watering hole. Footprints fossilize more than movement. They preserve intent, community, and sometimes desperation. Their positioning in layered rock allows paleontologists to reconstruct behavioral timelines over thousands of years. In Alaska's Prince Creek Formation, bones of juvenile hadrosaurs appear in abundance. Their small size and seasonal growth patterns preserved in the rock suggest that they hatched and matured in the Arctic rather than migrating. What's more, bones within these permafrost-locked sediments often show little sign of predation, leading some to hypothesize that top predators were fewer in these extreme regions, possibly due to the challenges of long winters with limited sunlight. The very rock that held these remains also traps a climate record, offering insight into the delicate balance these ecosystems maintained. Ankylosaur bones in polar deposits sometimes bear traces of post-mortem transport, such as polished surfaces or sediment-scoured pits. These geological signatures suggest the carcasses were moved by meltwater streams or flash floods before burial. The manner in which these bones came to rest adds a forensic layer to paleontology. Rock acts as both the crime scene and the witness. Some specimens appear crushed beneath the weight of collapsing sediment, hinting at sudden burial events, possibly during volcanic eruptions or glacial outbursts. Mineralization processes in polar regions differ significantly from those in arid fossil beds. The extreme cold slows decay, and permafrost preservation can even trap soft tissue structures for millennia. Iron-rich groundwater sometimes flows into buried remains, turning bones into vivid hues, deep reds, oranges, and purples that mark chemical interactions over millions of years. These colorful fossilizations help scientists identify how long a bone lay exposed before burial and the chemistry of its final resting environment. Rock layers also reveal the rhythm of life and death over eons. Cyclic sedimentary patterns in Antarctic formations show periods of flooding followed by drought, then growth, then ashfall. Each layer becomes a chapter in the story, and the vertical sequence reads like the rise and fall of empires. Occasionally, large gaps, unconformities appear, where entire epochs seem missing. These absences become mysteries of their own. Did a climate catastrophe erase the life above? Was it glacial erosion or tectonic uplift that stole those stories from the Earth? Some of the most tantalizing finds come not from obvious bones but from concretions, hard mineral nodules that preserve soft tissues, skin impressions, or even embryonic forms. These rock cocoons are often mistaken for ordinary stones until sliced open, revealing delicate structures inside. One such concretion in Australia held the partial remains of a small dinosaur embryo, perfectly preserved in pose. Its rock cradle acted as both tomb and time capsule, offering a fleeting glimpse into polar dinosaur reproduction that is otherwise all but invisible. The rocks of polar regions hold a legacy more than biological. They hold the memory of resilience. These ancient landscapes whisper the stories of creatures who thrived where few others dared. In each sedimentary fold and fossilized footprint, a testimony remains that life, even in the harshest environments, leaves a mark. As ice melts and more of these ancient layers are revealed, the legacy locked in rock continues to grow, reshaping what we understand not only about dinosaurs, but about survival itself.